so on. Also, because we are using long reads here, we are able to understand we are able to read the whole gene directly if you're able to design specific primers or something like that, where the whole gene gets amplified in one go and gets sequenced. So you do not have any miss outs in between because of the fragmentation which you're going through in case of any short read platforms. Applications specifically in terms of rare disease, because that is what we are trying to actually explain in this particular presentation. So large deletions is something which is very easily detected using long read platforms. Any rearrangements that you have, any duplications that you have is something which is very easily understood using long read platforms in the rare disease context. So if there are any specific disorders which has repeat expansion regions, so Nanopore is able to understand that very easily, because we are not having any PCR bias kind of an issue, because 99% of our libraries do not have any PCR amplification steps, as is the case in any short read platform, where you have to go for a compulsory PCR amplification step post the initial library preparation. So because we are also able to understand and have information on intronic variants and any specific promoter level disruptions, so we are able to capture more information and do not have negative false negatives that we might sometimes see with whole exome sequencing. How does a typical Nanopore library sequencing workflow look like? Basically, you will have your sample already available. The native DNA or native RNA is something which we can sequence on Nanopore without any PCR cycling or cDNA conversion. So once you have that available, you basically have the samples QC'd and everything, and then you go forward for the library preparation using multiple strategies depending on the sample type. Once you're done with that, you go through the sequencing process, which is again a real-time sequencing. This is the only platform in the world which allows you to do real-time sequencing, which basically means the amount of data that you generate, the amount of data per sample that you generate, the quality of the data you are generating, all of these information can be obtained in real time, and you do not wait for any post-processing of your data. Typically, you do not need to wait for 3-4 days after your run is complete and that get the information. You're able to get all this information from the very second minute of your sequencing run. So we are also having GUI-based data analysis pipelines as an offering along with every sequencer. So you do not need to have any command line knowledge or bioinformatics scripting knowledge. You basically generate your FASTQ data. You tell the analysis software, this is the location of my data and this is the analysis that I want to do. You click on start and it generates you the HTML report along with the files necessary for your tertiary analysis like VCF files, BAM files, and so on. Come to the cost and accessibility advantages that we have in case of Nanopore. We have the smaller range equipments to the largest high throughput instruments from Nanopore, and fortunately this is also the only sequencer that has gone to space, and there's a MinION which still exists in the International Space Station and is used for sequencing samples collected directly from the space. So we have flexible throughput options, like if you have a low throughput, with respect to samples, if you have like maybe 5 samples in 5 days or something, we have smaller devices to ultra high throughput instruments where typically you would be sequencing 24 human genomes every 3 days. So we have all those also covered, and because our instruments are small and you need minimal IT infrastructure for it to work with, basically one laptop connected to a sequencer works in any setup. So basically you have your library. If you have a remote lab that you already, a mobile lab that you have, you can prepare your library there itself and load the sequencer and start generating data in 10 minutes or so. So the strategy here being basically when we're talking of rare diseases as a context, so we basically try to promote Nanopore as a complementary technique along with whole exome sequencing. So this basically helps you to have a much greater yield in terms of whatever pathogenic variants that you're trying to understand and so on. With respect to integrating the whole setup in your pre-existing lab, because the infrastructure that we need is a very same as a very basic molecular lab. So you do not need any specialized setup, and you just add the nanopore sequencer, the reagents, and a laptop maybe, and that itself completes your setup. Yeah, I just have one slide here. Yeah. So conclusions and takeaways. So I won't spend time on this because we are already running short. So this is our whole product line that we have. We also have a booth out here, so please visit us, and then we can take this forward with respect to any discussions that you would want to do. And that's it for my presentation. Thank you.
Thank you so much, Mr. Saikat. Thank you so much. Ladies and gentlemen, we now take a short tea break of 10 minutes. Parallel sessions will commence in all halls. After the tea break, please consult your conference booklet for the hall allocations and kindly be seated in your respective halls after 10 minutes. Thank you so much.
Using the immunogenic protein of Mycobacterium tuberculosis for improving long-term memory response. So as we know that Mycobacterium, it infects humans and through aerosol media, and we can see that once it has been internalized by the host body, then it goes to the lungs, and in the lungs it forms, it activates the immune system, it brings in the immune cells, and at the site of the infection, a granuloma-like formation takes place. So once the granuloma disintegrates, then the mycobacterium can move out of the infected individual, and it can go back to the next human. In such a process, at least one infected human can transmit infection to at least 10 uninfected individuals. So with this graph, I would like to point out that with the current drop in the TB rate, there is no point that it can reach the end TB goal by 2035. So innovative steps need to be taken, wherein by around 2025 there was a decrease of around 10% decrease in the TB, and that is the reason why that if we want to bring it to by 2035 to zero, then there should be a decrease of around minus 17% per year. So that is the reason why we really need to speed up the process, and that is the reason why repurposing of existing drugs or vaccines come into picture. So the work started like 10 years back from our Professor Hasna's group, and they identified, they compared the non-pathogenic or opportunistic and totally pathogenic mycobacteria, and they found out that what are the different genes that are unique to MTB. So we followed on that work, and we came up with 25 sequences which we followed up, and out of that 25 we chose 4 which we annotated them as signature sequence because they were present only in the MTB. Interestingly, this signature proteins, they exhibit serospecificity in PTB patients. So that's the idea came that whether we can use it as a vaccine candidate or not. So we started the work for characterizing these proteins, and we are showing the data for one of the protein 1507A. So we infected the macrophages with purified protein, and we tried to assess the cytokine levels, and we found that it is pro-inflammatory in nature. Then once the macrophage had been sensitized with the purified protein, we again infected it with the recombinant M. smegmatis expressing this gene because we were lacking the BSL-3, so we used M. smegmatis as a surrogate strain. So again we tried to look for the cytokines, and to the left of the panel we can see that there is increase in the pro-inflammatory cytokines, indicating that this protein is highly immunogenic in nature. So the next thing that we did was that we infected the mice with the recombinant strain of M. smegmatis, and we took out the splenocyte, again treated with purified protein, and we looked for the immune profiling of the various sublymphocyte populations, and we saw that there is an increase in the significant increase in the CD80, CD86 and CD69 cells, which are important for stimulation and co-stimulatory markers. So we followed up this study, and we found that in mice, upon treatment with this protein or with the M. smegmatis, we took out the splenocyte, and we found that there is an increase in the IFN gamma and TNF producing cells. And interestingly, when we take the bone marrow derived macrophages and we add purified RB1507 protein, and we after 7 days of treatment, if we again infect it with MTB, there is a high uptake and clearance of the MTB as compared to the control cells. So then we, apart from the different markers that we are looking, we also try to look for whether there is what is happening to the memory markers. And we found that whenever the mice is infected with either the protein or the recombinant strain, we find that there is upregulation of CD44, CD62L markers, and that actually gave us an indication that this protein plays an important role in memory response. Now, if we look into what happens after 30 days of infection, we find that in the lungs, in the mice, it was leading to the infiltration of a lot of the immune cells. So with this idea, we then characterized all the SP proteins, and the stage was set for us to look for the effect of this protein on recombinant BCG. So since we know that BCG is the only vaccine, but the problem is that it doesn't mount memory response, so it fails to elicit protection in adult individuals. So we electroporated this gene in the BCG, and after doing selection recombinants, which is a patented process from our lab, and we do sequencing and quality check. So these are the various parameters on which we can assess the efficacy of the recombinant BCG, and these are the 9 parameters on which we actually test the efficacy of our BCG. And the first thing was that we took the PBMCs and we treated with the BCG, and we look for the antigen presenting cells. We get the CD14, CD16 cells, which are actually non-classical macrophages. And if we see that there is expression of CD86 indicating that macrophage activation is taking place. If we further go ahead and co-culture it with the CD3 autologous cells, we find that there is an upregulation of Th1 cytokines, indicating that the recombinant BCG is actually eliciting the immune response. So the next thing was that we wanted to see what is the effect of this recombinant BCG on memory T cell development. And in the in vitro system, we took the PBMCs and we infected with recombinant BCG. After 5 days, we removed the media, and after 10 days, when we look for the different markers, we find that there is an upregulation of T follicular helper cells, which indicates that there is enhancement of the germinal center formation. And the frequencies of the memory CD4 and CD8 cells also increased. So that gave us a good indication that our recombinant BCG is actually working in the lines of improving the memory response. So this was in vitro, and we followed up the study to see the different genes that are upregulated and downregulated by this, and we found that the RNA sequence heat map actually shows that there is differential expression of apoptosis, cytokine, and autophagy-related genes. And we carried out the in vitro studies to further validate this entire setup. For the interest of time, I'm just moving ahead. And the next thing was that whether if we give it to the mice, what is the response? A short-term response and a long-term response. So we look for the response of this BCG after 90 days. So when we gave this BCG to the mice, we took a strategy to give it by intratracheal method, because at that time there were some papers which showed that mucosal immunity is improved by giving it intratracheally. So we tried to look into that parameter, and we found that there is the frequency of T-rex is actually decreasing, which is actually a good indicator, because T-rex upregulation is actually proceeding to TB. So because of the reduction in T-rex, it was a good indicator that it was working fine. And then we followed it up with re-stimulation with MTB lysate on the splenocytes, and we found that cytokine stimulation is fine. So to sum up the year, we also looked for whether there is an increase in the trained immunity or not, and we looked for when we stimulate the cells from the mice, once it has been immunized, we stimulate it again, we find that there is upregulation of lactate, which is an indicator of trained immunity. And there is upregulation of different bone marrow derived cultures. We found that heterologous treatment of LPS or MTB lead to increased different types of cytokines. So further we looked for the antibody response, IgA, IgG, in the bronchial alveolar lavage, and it was giving a positive indication that it is mounting memory response, and it is the antibodies are well in line with what we are expecting. Finally, we looked for whether the BCG can promote central and tissue-resident memory cells. As we know, tissue-resident memory cells are the first line of defense whenever there is infection. And in our studies, we found that both the central and tissue-resident memory T cells has increased significantly, indicating that our BCG is working fine. We have tested this on a pilot study to see what is happening if the immunized individual is again given an infection, and we found that there is clearance of bacterial infection whenever the individual has been immunized. So these are so to sum up, we are at a stage where we have a project wherein we are looking for the efficacy. We are at a technology readiness level 4-5, and we have to move to 6, and we expect that we will carry out these experiments on guinea pig models. And once that has been done, so this is one of the another aspect that we are doing. I just showed you that we had worked with one of the protein, but we have characterized 4 signature proteins. We have developed a multi-epitope vaccine consisting of the important epitopes from all these 4 proteins, and the multi-epitope vaccine is working fine. We are optimistic that we can carry forward this study in animal models. So I'd like to thank our group. This is the overall work that we do. I would like to acknowledge Department of Biotechnology, Indian Council of Medical Research, Department of Health Research, DST Imprint, and Council of Science and Technology, Uttar Pradesh. And I would like to thank our collaborators, Professor Syed Hasnain, Professor Nasreen Ehtusham, Dr. Siva Kumar from NIRT Chennai, Dr. Mitra from AIMS, and yeah, thank you. If there is any question, I will be happy to answer.
If not, then we can move on to, yes, please. Only one.
Pardon? The concentration was around 4 microgram that we used. And the second one? BMDMs are bone marrow derived macrophages. So we used it because it is a better indicator of what we get in terms of cytokine response, because generally we use raw cell lines. So once we have completed those studies on raw cell lines, then we move to BMDMs, and BMDMs actually give us the real picture of the cytokine profiling.
Thank you. Thank you. So you may have a further discussion during the lunch. So our next presenter is Professor Monica Sharma, who is a professor at Miranda House, Delhi University. So she'll be talking about a multiple epitope vaccine derived from late stage PPE proteins of tuberculosis, induces strong immunogenic and memory responses. So over to you, Professor Monica.
Good afternoon, everyone. I would like to thank the organizers and would like to start and present my work which we are doing on an epitope vaccine. The vaccine which we have derived is a multi-epitope vaccine. My colleague has just described about he has patented one of the epitope designed vaccines. We have already designed it, and now we are in the process of validating its immunogenicity and protective efficacy, both in BALBC mice and PBMC derived from healthy individuals and PTB patients. I would like to skip all these basic slides because we have already been listing about the importance of TB as a disease and requirement for the vaccine to be developed for it for controlling it. So there are many vaccine platforms which are under clinical trials, and we are interested in subunit vaccines because we believe that subunit vaccines are much safer and easy to handle, though the immunogenicity is considered to be low, but that can be taken care by using various types of adjuvants. these are some of the subunit vaccines which are in clinical trial. And the most promising, which is in the phase 3 clinical trial, is M72AS01E. And it has an interesting component, that is PPE18. Because in our lab, we, along with the DOSA regulon, which Sadhna ma'am yesterday described, that we work on 2 different families of proteins, DOSA proteins, which are dormancy-associated antigens, and PEPP proteins. So we have been extensively working on PEPP proteins, and we have found that most of them are uncharacterized, and they're very unique and interesting proteins. 7 to 8% of these, the genome of MTB is dedicated to code for these proteins, and we have been scanning the individual proteins. And another interesting aspect about these proteins is their differential expression. At different times of infection, they express. Some of the proteins express at early stage, some at 30 days of infection, some at both 30, 90, and some exclusively at 90. So this has always fascinated us. So what we did was, because most of the vaccines, they are mainly focused on the early antigens. So we thought that why not to take antigens which are expressed at the chronic stages of infection and combine them with the early stage genes. So we did this immunoinformatics study, and it was published in European Journal of Pharmaceutical Sciences, where we scanned all the late-stage expressing proteins of PEPP family. And then we isolated 1,466 promiscuous epitopes, which were binding with more than 4 MHC alleles. And then from them, we segregated 469 cross-reactive. Because interesting thing about PPP proteins is that they have homology sequencing. So if a epitope is from one particular protein, it can be found in various other proteins of the same family. So we segregated those epitopes and called them as cross-reactive. And then we scanned for their antigenicity again, based on immunoinformatic tools. And in total, taking all these 3 parameters in consideration, we isolated 42 promiscuous epitopes, which were immunogenic as well as cross-reactive. Then out of these 42, we took the top 10, and did all the analysis. We call them PCAPs. And so I'm skipping all these slides. Then we computationally designed this construct where we have used adjuvant as ESAT6 is an early secretory and extensively studied and found immunogenic protein of MTB ESAT6, which we took as an adjuvant. And then those 10 peptides we took one by one and linked with linkers. Then all the immunoinformatics study showed that this particular vaccine construct protein was able to specifically bind to TLR2, and through immune server, we could find that it is capable of generating immune response, but we needed to validate it. We purified this protein, characterized it, and then we used this immunization strategy where we took PBS as control, only PPE-VC, as we called our protein vaccine as PPE-VC. Here, PPE-VC was given to mice. One group was given with PCG as a control. Then we use co-immunization, homologous prime boost. PCG plus PPE-VC together was given as prime, and boosting was also done with this same combination. Another group was given with PCG as prime and boosted with PPE-VC, R protein. And then another group was primed with BCG plus PPE-VC and then boosted with PPE-VC. So all these groups were immunized, and the immunization schedule is here. And so 8-day euthanization was done, and we found these results. As we can see, very good immune response was generated, IFN gamma producing CD4, CD8 T cells, and in splenocytes and lungs. And here we can see memory cells, effector memory, good immune response in terms of effector memory and central memory cells, both in lungs as well as in splenocytes, CD8, CD4 both. And here we can see that interesting part is that the frequency of the Treg cell was reduced. Expansion of Treg cell was reduced. Dendritic cells were studied for the activation, and we can see that dendritic cells were found to be activated, both dendritic cells derived from splenocytes, from lungs, as well as from bone marrow-derived macrophages. And we also observed the activation of macrophages following immunization. Same immunization schedule was used for all these studies. And if we talk about the cytokines, again, we found elevation of cytokines in response to our vaccination. The best response was observed in all the cases. It was in the heterologous prime boost strategy. Though our PPVC was alone also was quite capable of inducing good immune response. We also studied the TLR signaling pathway. Again, it was found to be very elevated and significant on immunization, both in lung cells and bone marrow-derived dendritic cells. So this was the work which we have done till now. We have started with the protective efficacy because that is the most important part that needs to that needs to be done to confirm the protective efficacy that we are doing, and the data is not presented here. Then we also did the immune response generation in the patients, healthy as well as patients of TB. So till now we have enrolled 18 PTB patients and 30 healthy individuals. And here we can see that in response to stimulation with our vaccine protein, there is a significant increase in the both CD4 IFN gamma producing cells and CD8 IFN gamma producing cells in both PTB patients as well as in healthy individuals.
Professor Sharma, you
another
have 2
important
minutes
aspect is the IP10, which is also interferon gamma, CXL10. It is again a very important biomarker for the Th1 type of immune response that we have found. It is also elevated in all our subjects in response to our protein. Then we also look for the PD-1, and interestingly, if IFN gamma and PD-1 co-expression was found to be elevated. But the most important part here I would like to mention is that the PD-1 is supposed to be, everyone says, oh, PD-1, that means T cells are going for exhaustion. PD-1 are also considered to be activation markers, and along with interferon gamma, if they are there, and we can see there that in PTB patient, it's not increasing significantly with advent of time. Though we have done till 72 hours, but it's not increasing. So it's indicating that the PD-1 here expressed are not as exhaustive markers. Rather, they are the activation markers along with the interferon gamma expression in the T cells. So we looked for the memory cells. Again, our vaccine protein, the healthy individuals as well as PTB, they responded to the protein and there is an expansion of memory cells. Similarly, we looked for the activation of CD4. So CD4 and CD8 both were found to be activated. And regulatory cells, there is no significant expansion of regulatory T cells in response to our protein, which is an interesting and very important aspect. So it tells about the balanced immune response that can be generated by our protein.
You may conclude your talk.
So as a conclusion, I would like to mention that this multiple epitope vaccine till now, what we have found is it is found to be a very promising subunit vaccine, and we need to further validate its protective efficacy. But yes, we hope that it can emerge as a good vaccine candidate in future.
Thank you
so
Sharma.
much. I would like to acknowledge my mentor, Professor Sadhana Sharma, my collaborator, Professor Anita, Dr. Parul, she's the clinician, and Professor Garima Khare from University of Delhi.
Thank you, Dr.
Sharma. Thank you so much.
So, if we have any burning one question, otherwise we can move on to next presentation. No.
Yes, sir. Sir, for PBMC, it has to be in vitro and Balsi mice in vivo. Yes.
Thank you. Thank you.
Okay. Thank you.
May I now call Dr. Mithilesh Kumar Singh, who is a senior scientist at SCICC, right?
he will be talking about trained immunity in chicken.
So good afternoon to everyone. Thank you organizers for giving me this opportunity to present in this Immunocon 2026. So the topic of my talk is exploring the trained immunity in chickens using sulfated beta-glucan and Newcastle disease vaccine. So as we know that immunity is of two types, innate and adaptive. As innate is non-specific in nature and it acts very rapidly in comparison to the adaptive immunity, which has a key attribute of specificity and memory, but it progresses quite slowly in comparison to the innate immunity. So despite having despite lacking the adaptive immunity in plants and invertebrates, they still show a kind of memory response. Once there is induction of immunity, there is kind of memory response. So this sparked a curiosity among the scientific community that how this memory is generated despite lacking the adaptive immunity, because we know that the key attribute of adaptive immunity is the memory. So recently, last decade, the concept of this trained immunity, or this is also called as innate immunity, this was been introduced by the Natia and Nitia et al. And they referred it as a process where the innate immune cells, like mostly it is monocytes, macrophages, NK cells, or even dendritic cells, so which get trained with certain stimuli or certain agonist. And after that induction, this process is called as priming. So there will be a phase that is called as resting, and subsequent to that, if there will be any exposure, whether it is related challenge or unrelated challenge, so there will be a quite heightened immune response. So heightened immune response is named as trained immunity or innate memory. But another possibility may also be there that despite instead of this heightened immune response, there could be decreased immune response, or immune response may be towards maybe at the basal level, and that is called as tolerance. So it all depends on the species and the agonist, what is being used for the training or priming of the innate immune cells or the individuals. So basically this innate memory is involved. There are two processes which are governing the induction of trained immunity. One is metabolic reprogramming, and then another is the epigenetic changes which occurs in the innate immune cells, which leads to the reprogramming and then activation of the cells after re-stimulation or challenge with the related or unrelated pathogens. So the concept of this trained immunity has evolved recently, and it has opened the new era for the development of some novel vaccines and then adjuvants, and basically receptors which are involved in the induction of this trained immunity. So it is not like receptor, again it's a kind of PRR, and then C-type lectin receptors, that is dectin-1, which is mostly involved in the induction of innate memory. So once there is priming, so followed by a kind of phase that is called as resting phase, and then once there will be induction with the certain stimuli, may be related or unrelated, so there will be quite heightened immune response that is called trained immunity. And the ligands which are known to induce this trained immunity is mostly it is BCG vaccine, as our previous speakers they have discussed. So it is mostly BCG, and it has explained quite long ago that BCG immunized individuals have protection against many infections which occurs concurrently, like mostly in neonates if you see. So mostly respiratory infections, the degree of infection will be quite low in BCG immunized individuals in comparison to those who are not vaccinated with the BCG vaccine. So in food animals, if you see, the vaccination with the BCG is not recommended, or it is prohibited because of the diagnostic interference. So the other option which is left with is the beta-glucan, that is a kind of polysaccharides, and these beta-glucans have been quite widely used both in mammals and avian species for the immunomodulatory studies, but its studies related with the trained immunity induction is quite restricted. Besides this BCG and beta-glucans, there are several other molecules like flagelline, LPS, poly-IC. Those are also being explored for their trained immunity potential. So basically in animals, if you go for oral administration, that is quite convenient, but at the same time we have to administer for a long period to induce the trained immunity. And parental administration, suppose if you are going for even once, so that may be equally effective. And the problem with this beta glucan, it is water insoluble. So suppose if we are able to modify it somehow to make it water soluble, then there could be quite desired immune response in terms of induction of trained immunity. So in any species, like suppose if you see the neonatal stages, so there you may find that individuals are quite vulnerable or susceptible to several infections because of the lack of adaptive immunity. And at that stage only there is innate immunity and the maternal antibodies. So definitely at that point of time, suppose if we are able to strengthen the innate immune responses by harnessing the potential of trained immunity, that could be a quite promising option. And what I am saying, like harnessing the trained immunity potential by using the beta glucans, and that too by modifying it so that it could become soluble. So definitely this could resolve the issue of immunomodulation, especially in animals. So we have taken two objectives for this work. First was the extraction of beta glucan and its modification by adding the sulfates so that it could become water soluble, and then it has to be evaluated under in vitro system for its trained immunity induction potential. So we have taken the specific pathogen-free birds, and then source of beta glucan, we have taken the commercially available dry yeast for the extraction of beta glucan, and then as a positive control, we have purchased standard beta glucan from commercial sources. And then we have used the acidic extraction method for the isolation and purification of the beta glucan, and then it got modified by adding the sulfates using standard methods. And then we have characterized it by knowing the carbohydrate and protein contents in that, and then its molecular vibrations by FTIR. And in second objective, once we got the beta glucan, in-house prepared beta glucan and sulfated beta glucan, then we went for inducing the mononuclear cells of chicken for knowing the induction of their trained immunity potential. So this was the methodology where we have given the beta glucan, means in-house prepared, then sulfated one, then commercial one, and besides that, one widely used vaccine which is being used in poultry, that is Newcastle disease vaccine. So we have primed the cells, monocytes, on day 0, and then we kept resting for 5 days, and after that we have given 2 heterologous stimuli. One was the TLR2 agonist, that is PAM3CSK4, and then another was the TLR3 agonist, that is double stranded RNA, that is NDV vaccine. And then we evaluated the immune response, especially the hallmark cytokines which are being expressed in trained immunity at priming stage, then resting stage, and then re-stimulation or challenge stage. So these are the findings, like carbohydrate and protein contents. If we have once we have compared with the commercial one, so that was in the acceptable limit, and even protein content was also under the acceptable limit. Under FTIR spectroscopy, we found the typical beta glucan peaks as earlier reported, and even sulfate groups was also being found as per the previous reports. So once we primed the monocytes with the beta glucan or sulfated beta glucan, so during priming stage, we found that there was quite high proliferation or high fold change expression of the 3 inflammatory cytokines, IL-1 beta, TNF, and IL-6 by all the agonists in comparison to the control. And once we have given the heterologous stimuli like PAM3CSK4 or this NDV vaccine, we found that there was quite heightened immune response after 6 days of re-stimulation. So this confirms the immunostimulatory potential of beta glucans, and among all the beta glucans we found quite heightened immune response with the sulfated beta glucan. Even nitric oxide also we have estimated, because this is also a key molecule induced in case of trained immunity, and that was also being upregulated with the sulfated beta glucan training or priming. And this hypothesis or these findings were further confirmed by using the 2 inhibitors which are known to inhibit the metabolic and epigenetic changes, which are the key pathways in induction of trained immunity, and that further confirms or validate our findings that yes, what the stimulation or induction is happening because of the beta glucan, that is operated through this two pathways, that is epigenetic and metabolic reprogramming. Similarly, besides this inflammatory cytokines, we have checked this nitric oxide production and then its inhibition by these two key inhibitors. So summary and conclusions. In brief, we have proven this in vitro studies by using the modified sulfated beta glucan that was found to be quite effective in induction of trained immunity. And we are carrying out this work to further validate under in vivo system, and subsequently this could be used as a potent adjuvant by optimizing its dose and then its delivery system. Thank you so much. Thanks to all my students and the DBT for funding this research.
Thank you for a great talk. I think we don't have that much time for a question, so we'll move to the next speaker, please. Dr. Shivshankar Kumar Singh from Scientist F, ICAR, PM or IMS Patna.
Again. Good afternoon. First of all, I would like to express my heartiest thanks to the organizer, particularly Dr. Dilafruz and his team. Today, in this time, I'm going to discuss immunochemotherapy against visceral leishmaniasis, a multi-epitope PDI chimeric construct which can boost embryome treatment outcome. So, leishmaniasis is at the verge of elimination. The number of cases reduced to 400 from 40,000 in last 10 years. But even though there is certain hurdles on the way of eradication, that is drug unresponsiveness, HIV-VL co-infection, there is still no vaccine, and immunotherapeutics are still awaited. So critical issues are unresponsive to treatment, relapse, reinfection, and drug resistance. The degree of resistance can be understood by this cartoon, this picture. So this is hospital-based study to assess the degree of drug resistance. And you can see that out of 56, 24 was unresponsive to amphotericin B. And this parasite was resistant up to the degree 1.5 macromole, that is very hard for even for macrophages, host cell. So drug resistance in leishmaniasis is knocking out anti-leishmanials one by one. Once the successful drug antimonials is out of use, once the drug of choice miltefosine for Kala-Azar eradication program has been relapsed with single dose embijome, relapses are coming after embijome and even after combination therapy. So in last 125 years, that is the history of detection of visceral leishmaniasis parasite, we couldn't produce a single vaccine for application, clinical application. And issues for the vaccine are durability, antigenicity, and safety. So On the other hand, we can promote immunoprophylactic agents or immunotherapeutic agents which can support chemotherapy. Because for sterile cure, strong immunity is needed. In one of our study, we had observed that recombinant PDI, protein disulfide isomerase, prime dendritic cells triggers NF-kappa B signaling and was promoting pro-host TH1 polymerization, which was minimizing the LD infection. But this was dendritic cells, but not macrophages. In dendritic cells, it was promoting TH1 cells, TH1 type immunity and expression of nitric oxide, upregulation of nitric oxide, followed by the reduced spleen parasite. So we decided to pick up epitopes from this protein for chimeric construct. So we, with the help of different softwares of bioinformatics, we selected certain peptides for validation, and after validation, we selected some peptides for wet lab, and this is the MD simulation. This I will skip these slides. So first we used these peptides to see if these peptides are able to express or boost CD69 expression on T cell. So because that is the first activation marker and very good marker actually. So we used that and observed that many of the peptides was capable to express induce higher expression of CD69 for both restricted to class 1 and those restricted to class 2. So after that, those peptides were tested for in vitro assessment of cytokine induction, and we observed that some of the peptides were able to elevate hyphen gamma IL-10 ratio both in class 1 and class 2 group. So using that peptides, we formulated the chimeric construct for both class 1 and class 2 and evaluated in mice model to see if it can induce the protection using ambijome, without ambijome, or with ambijome. I mean, ambijome is the first line drug nowadays, and fusion construct with ambijome. So this construct, both 9-mer construct and 15-mer construct, was able to induce TH1 polarization and finally clearance of the parasite. So this is the safety aspect of this construct. We tested this construct for assessment analysis of ALT, AST, or alkaline phosphatase, and finally we evaluated whether this construct can reduce the spleen weight or not. So KFT was also validated. KFT kidney function test was evaluated, and you can see that even this construct is more safer than ambijome alone, and both for creatinine, urea, and uric acid. So fusion construct composed of human HLA works as an immunotherapeutic candidate in combination to LAMB, liposomal lymphotransfusion B, for treatment of visceral leishmaniasis, and it needs to validate in humanized mice because this is done on biopsy mice. Thank you.
So we have time for a few questions from the audience. Do we have any?
Yeah.
Actually, you skip the bioinformatics part, but I am a bioinformatician. That's why I am asking if you allow to me.
Mm-hmm.
Actually, you do the molecule simulation and dynamics study. So, on which platform you do it? On which software you have to do it?
Yeah.
GrowMax. And up to how many nanoseconds? 100 nanoseconds.
Yeah.
Okay.
So actually, this work was contributed by Amrita Singh and Dr. Manas Ranjan Dikshit sitting in the audience, and he was looking the aspect of bioinformatics part.
Okay,
So
and the
we are
docking
discussing
part is
it.
also you do the online tool.
Yeah.
Docking
Yeah.
is the online tool.
Yeah.
You may have a discussion during the lunch.
Okay.
So this is our team in background. Thank you.
Thank you, sir. So may now call Dr. Shrikant Sandhu. Is it a research scientist at SKICC? And we'll be talking about host immune reprogramming as a strategy against MDR pathogens.
Good afternoon, everyone. First of all, I would like to thank Immunocon organizing team and special mention to Professor Dil Afroz. So today I'm going to talk about how the host immune reprogramming helps as a strategy against MDR pathogens. We know that MDR pathogens are nothing but a global, it's a threat to the global. And there are the antibiotic discovery is going on, but still we need another strategy which is nothing but host immune reprogramming, which can enhance the host immunity to fight against infections. So I have used one of the infection model, which is Salmonella. So we need host-directed therapy because of their antibiotic tolerance and their MDR emergence due to the persistence of misusing or abundance of antibiotics are used. And therefore, we need immune-based therapeutics which can help to fight against different types of infection. And we have screened around 720 molecules, natural compound library. And we found that there are the 5 molecules which are potently inhibiting the Salmonella infection. And we have done the dose kinetic analysis. We found that even at the 0.1 micromolar, this functionally is the molecule which is significantly inhibiting the bacterial growth. And we asked whether it is showing any cytotoxicity to the cells, and we found there is no cytotoxicity. And we asked another question, is it directly killing the bacteria? And we had another experiment. We kept the bacteria and in presence of this drug, and overnight we have cultured, and we found that there is no decrease in the bacterial growth. So which means that it is not killing through the direct mechanism. And then we have checked what about the nitric oxide levels in this condition. We found that the nitric oxide levels were elevated in the FCN treatment group. And we have asked, okay, if it is the nitric oxide playing an important role, if we inhibit the nitric oxide in presence of this drug, in the combination of FCN plus NOI, will it decrease that or not? We found that when we have used the FCN plus NOI, the drug effect was mitigated in presence of this combination. And we also found the same thing, the nitric oxide levels were decreased in the combination compared to the FCN alone. And we done another experiment. If it is through the nitric oxide, we will do the nitric oxide, the NOS2 knockout mice we will use. The NOS2 knockout macrophages we have isolated. And we have checked in the same condition in wild type and NOS2 knockout mice. And we found that in the NOS2 knockout mice, in these macrophages, we haven't found the effect of the FCN on these salmonella infection, but we can see that the FCN, which is in wild type mice, wild type macrophages, we found that the FCN was significantly decreased the salmonella burden. So before going to the in vivo testing, we have done the toxicity of this drug in vivo. So we checked in the liver LFT, liver functional test, the main markers are, these are the ALT and AST. We found there is no elevation of these markers. And we also found in the KFT, which is kidney function test, we found that there is a creatinine and urea levels which are normal as comparable as the control. And we also done the histopathologic analysis of the vital organs like liver, kidney, and spleen, and we found there is no abnormalities in the tissue. Then we have done the in vivo analysis. So after the infection to the wild type mice and NOS2 knockout mice, at 48 hours we have done the we have done the imaging of these mice, and you can see that at 48 hours the salmonella was significantly they are make the groups in the intestine, and you can see that at the day 5th, in the presence of FCN in the wild type mice, you can see the bacterial burden was significantly decreased, but we haven't found in the NOS2 knockout mice. And it is also another thing is in salmonella infection, there will be shrinking of the colon. So we have checked what about the colon size. And the colon size was significantly as comparable as control mice in the wild type, but we haven't seen in the NOS2 knockout mice. And we also checked the bacterial burden in the feces, spleens, cecum, liver, and we found that the bacterial burden was significantly decreased in the wild type FCN, but not in the NOS2 knockout mice, which is treated with the FCN. And we also done the serum nitric oxide levels, and we found that the serum nitric oxide levels, which are significantly increased in the wild type, but it was not found in the NOS2 knockout mice. Then we have done, if it is nitric oxide increasing, we asked the question, what about the metabolites? What are the metabolomic analysis? So we have done the metabolomic analysis of the serum samples for all these mice. And you can see that these are the metabolites which play an important role in the nitric oxide generation. So these are the metabolites which are significantly increased in the FCN treated group. And these are the pathways which are significantly elevated in the wild type FCN mice, mice which is treated with FCN. And we found that yes, the nitric oxide related pathways which are significantly elevated and due to this FCN compound. And we asked another question, is it making any changes in the immune levels of these mice? And we found that there is a significant increase in the macrophages in the FCN treated group in wild type, but not in the NOS2 knockout mice. And we also found the M1 phenotype markers, which are significantly increased. And we have asked whether that there is a Th1 type T cells are increased or not. So you can see that in wild type mice, you can see there is a significant increase in the IF and gamma in these FCN treated mice, but not in the NOS2 knockout mice. And there is IL-10 levels, which are non-comparable. And we also found at the mRNA level of these Th1 related cytokines like IL-1 beta, IL-12, IL-6, and all these genes levels which are significantly upregulated in the wild type FCN treated mice, but not in the NOS2 knockout mice. So we asked a question, if it is working to the susceptible bacterial strains, will it work to the multi-drug resistant bacteria or not? So we got some of the multi-drug resistant strains clinically isolated from Dr. Bhautos Das from THSTA. And we have done the same experiment in these settings. And you can see that the Salmonella typhimurium, and we also done with the Shigella, is another multi-drug resistant strain. We used this compound and we checked whether these multi-drug resistant strains which are significantly inhibited or not in this FCN compound treatment group. And we found that which is significantly the colonies, you can see that they're significantly decreased in the FCN treated group compared to the infection. And we asked another question, is there memory responses or something different in these groups? And we found that the T central memory and effector memory levels significantly increased in this treatment group, which is significant, which is actually surprise to us. Then we have done another small experiment. We have given the pre-treatment of this compound at 14 days before, 7 days, and minus 0.1. And you can see that after the pre-treatment of this functionally, the weight which was rescued in pre-treatment group compared to the which is not treated, which is infection group. And the bacterial burden, which is also we found that significantly decreased in pre-treatment group, which means that there is a memory response which is elevating in this group.
We have 2 minutes left.
And the summary of this story is this group, which is this compound which is activating the nitric oxide pathway, and nitric oxide playing an important role in significantly inhibiting the FCN, and we found that there is a Th1 immune response was significantly increased, and the surprising thing is we found that there is a memory response which was increased. And with these data, we are doing some of the experiment which are very interesting results we got. We are taking these data further in the next story, how this memory response was increasing, and we are also checking with other bacteria like MTB and Klebsiella, and these are all these things we are looking into this one. And I thank our department head, Professor Amit Awasthi, and our team members. And thank you very much. And looking for collaborations if very interesting. Thank you.
Thank you for sticking to time. So we have time for one question, please.
So the experiment which we are planning, I said, so we are going to look into all these things. Yes.
May I?
Yes, that goes.
Hi, this is Rajpal.
Yeah, yes.
Looking into the AMR concern and all these things, we are struggling with pathogen diversity and all these things. If we move to host-directed thing, so there is more kind of a changes and different host factors are involved in all these things. So do you think, if it works, it's fine, but do you think it is practically looks possible in next 3-4 years?
Yes. We are mainly, we are relying on antibiotics only. And we are not looking into our host machinery, which is more important to kill these and fight against these bacteria, whatever it intracellular, extracellular. So we should look into this host directed also, because if our host immune reprogram is become suppressed in these conditions, you have to activate, you have to enhance. So it will help to fight against. So this is one of the, nowadays it is a growing field in this one.
Thank
not
you.
only the antibiotics, you have to look into the host reprogram.
I'm sorry I had to cut short you.
Yeah, sure.
So we can. Yeah. So our next presenter is Professor Hridesh Prakash, who is a professor at MIT University. He will be speaking on sphingolipid mimics, a promising host-directed therapy for controlling mycobacterial burden in host. Over to you. Welcome in, Professor.
Thanks.
So first and foremost, I would like to thank the organizer for giving me an opportunity to discuss our work that we are doing in our lab. So I come from the MIT Center for Translational Research, which was established 3 years back in the MIT University. So the mandate of the center is to develop, next generation immune pharmaceutical and immune therapy for the TB, cancer, and IBD. So these are our current verticals that we are working on, and we are investing heavily on development of host-directed approach against tuberculosis and developing CAR macrophages like CD47 positive and negative CAR macrophages against solid tumor. And we are also, developing the stem cell and macrophage-based immune conditioning approaches for the AGVHD in the AML patients, which are developing sepsis. So with regard to the academic program, we are running PhD in translational immunology, first time in country, and starting MSc in immunopharmacology from July. So now coming back to my talk, I would not touch upon this because a lot of work have, the set has already been set by the previous speakers. So what is so important about our work, we are targeting and using the sphingolipid as a host-directed approach because sphingolipids are the special category of lipids which are involved in hashing the bugs, and maintaining the immune homeostasis. So once they get perturbed, they lead to they develop and they are involved in the pathophysiology of various diseases which are indicated here. But for keeping our focus on tuberculosis, so we are we are proposing them as a dual specific drug target for controlling pulmonary infection, mainly the tuberculosis. And in this context, we know that sphingolipids are the toll plaza for the pathogen. When I mean toll plaza, I mean they can control, they can trap, they can patrol all kind of pathogen once either both at the entry or as well as intracellularly. So there are a couple of evidences that sphingosine kinase is responsible for mycobacterial replication and dengue, chikungunya, meningococcus, cryptococcus, and many more. So this figure is clearly showing that the sphingolipids have a potential for controlling the mycobacteria if tweaked properly in the patient or in the animal model system. So we have demonstrated back in past that mycobacteria uses the sphingosine kinase for the entry and then subsequent activation and causing the inflammatory microenvironment. So taking the message from this, we have published this is cutting the long story into the short. We have shown that sphingosine stabilizes the iNOS expression in the macrophages, and it controls the mycobacterial infection in the macrophages by promoting the LAMP2 acquisition and their activation. It stabilizes the VDEC and VATPase protein in the early and late endosomal compartment, and this controls the P38 and NF-kappa B pathway in infected macrophages, right? So now the question is whether it can control the pathogenic mycobacteria or not. So with this, we set the clinical relevance in our setup and we know that poor gradient of the sphingolipid in EPTB and NRTB patients is contribute to the poor immunity. And the key question here is whether sphingolipids would be effective against controlling pathogenic mycobacteria or not. So with this, we actually did our preclinical studies using H37RV aerosol challenge method and by supplementing infected mice with a sphingolipids mediator, both in prophylactic and therapeutic setting. And we have monitored the tubercular burden and vis-a-vis immune monitoring. So our first experiment clearly shows a crosstalk of S1P with a interferon gamma for promoting the M1 phenotype in the naive macrophages. So basing on this, we did the infection experiment in the raw macrophages in presence of S1P, and what you can see that S1P is nicely maintained high titer of interferon gamma, nitric oxide, but it's able to control the IL-6 burden. So that was very nicely correlated with the CFU analysis in the raw macrophages. So this clearly indicates that tweaking sphingosine 1-phosphate pathway will favor the host in controlling the mycobacterial replication. So with this, we launched the first animal challenge studies at Jalma Agra, and we used the aerosol-based method and used the prophylactic and therapeutic conditioning of animals with the S1P, purified S1P, DHS, FTY720, and supporting our hypothesis, what we clearly see that therapeutic administration of infected mice with the S1P or FTY720 both in prophylactic and therapeutic setting, enhanced the sphingosine 1-phosphate level. So it's indicating that, this intervention was sufficient to cause the enhancement of, serum or pulmonary S1P titer. So accordingly, this increased titer were able to control the mycobacterial burden in their lung at both day 17 and day 31 post-infection. So this was kind of convincing to us. Now looking at into the lung micromilieu, we realize that Mycobacteria tuberculosis hijack the S1P receptor 3. For those who don't know about the sphingosine 1-phosphate receptor signaling, S1P signaling, so I want to mention that S1P has a 5 receptor, 1 to 5. S1P in a normal setting, uses S1P receptor 1, while S1P receptor 2 and 3 remain silent. So here in this context, what we realize that tuberculosis target S1P receptor 3, which is the immune receptor associated with the Th1 immune responses. So what we clearly see that, the hijacking of Mycobacterium tuberculosis for S1P receptor 3. And interestingly, what we realize following our hypothesis, S1P potentially enriched the alveolar space with S1P receptor 3 positive macrophages. That means, S1P treatment in the infected mice, enhance the infiltration of S1P receptor 3 CD4, positive macrophages as evidence of, improved innate immune defense in these mice against the pathogen, and, adding to the further mechanism, So, for that, to confirm whether S1P receptor 1 would also be involved, or would it, temper the innate immune responses? So we treated them with the S1. We infected the mice in presence of FTY720, which is a structural analog of S1P, competing the S1P with its binding to the receptor 1. And here also we clearly see that, if we tamper the receptor 1, so it also enriched the receptor 3 signaling in the macrophages, suggesting that S1P uses the S1P receptor 3 in controlling the animals. So following this, we further analyzed the interferon and IL-6 titer, and like before, we clearly see a positive correlation of S1P to the interferon and negative correlation of S1P treatment with IL-6, supporting the protective immunity in the animals. So further, we analyze that S1P potentially skew the pro-inflammatory programming in the infected lung. And that, so this came to this model that S1P is potentially anti-tubercular in nature and can be used in clinic for the management of tuberculosis. Now, at that time, we were excited, but then when we sat with the clinicians, I realized that this cannot go to the clinic. So there's a roadblock here because S1P is a purified lipid and has an allergic and autoimmune manifestation. Also, it has a very strong impact on lymphocyte egress, so it cannot be used, because S1P
Prakash, you have 2 minutes.
Yeah, S1P is a chaperone for the C5a, which is a very strongly, strong activator of mast cells. So that further add to the reservation of its uses in the clinical setting. So we come up with a strategy. An alternative strategy, that is by using the pharmacological approach, and that here we are using the stimulatory approach by using L-serine and palmitoyl coenzyme A and sphagnine in our approach. And what we realized that, L-serine potentially killed exponentially growing mycobacteria, and this uses the sphagnine as a first sphingolipid metabolite. And that L-serine is able to kill the mycobacteria in variety of the macrophages we have tested. Now we have realized that L-serine also reduces the IC50 value of first and second line anti-tubercular drugs against in killing the one-directional killing of the mycobacteria. This we have tested with the REMA analysis. And now when we challenge the raw macrophages with the H37RV, we realize that L-serine at a very high dose here is able to control the infection on its own, and it was able to work as an adjunct for the anti-tubercular treatments. Same was the case with the THP1 human monocytes. And then, surprisingly or interestingly, what we realized that S1P and palmitoyl coenzyme A was able to control the clinical isolates of 2 clinical isolates, from one isolated from SN Medical College Agra and second isolated from
Prakash, you may
AIIMS
conclude your
with this message, we challenge the animals with the L-serine. We challenged the animal with the MTB in the presence of L-serine, and there also we saw a very clear adjunct role of L-serine and palmitoyl coenzyme A. And interestingly, what we realized that L-serine was able to control the or normalize the pathology of the lung, which was severely infected with the mycobacteria, dropping down the pathological score from somewhere around 12 to 0. So it means it is a very strong and potential intervention against the tuberculosis. So we then actually went ahead and analyzed the impact on the human macrophages. And then, so because of lack of time, I have to skip this. So what I could clearly see that, L-serine was also able to disrupt the foamy macrophages, foamy phenotype of the macrophages, which is associated with the MDR pathophysiology. And that, with this we approached the ICMR, MedTech Mitra, and they also gave us a green signal that we can use this as a nutraceutical against tuberculosis. And I would like to tell here that this will be the first TB specific nutraceutical of country. And now we are heading for our phase 2 clinical trial. This is our trial protocol, and our major, intervention is whether it is able to reduce the sputum conversion in both DS and DR cohort of the tuberculosis, and that we are now going to invest. And I would like to thank all my group member and my collaborator, and here and something from funding, and thanks a lot. Yeah.
So I could see one hand raised, so we may have one quick question.
Yeah.
Thank you, Dr. Prakash, for such an insightful talk.
Yeah.
You mentioned L-serine was used as a higher concentration and that it sensitizes the bug to killing by rifampicin. So then how can it serve as an adjunct therapy if it is used at such a high concentration?
So that's a good point. Because of lack of time, I could not show you our PK/PD analysis. So what we realized that, with the presence of L-serine, the rifampicin degradation is also reduced. And it is able to reduce the IC50 value of rifampicin. And if you go to the mechanism, see, any host-directed therapy work on a multifactorial ways. There is no single mechanism which is enabling a therapy to control the treatment, be it cell line or animal or a patient. So we believe that it is sensitizing the impact of rifampicin by stabilizing the ROS, stabilizing the antioxidant, because L-serine, when get metabolized to the L-glycine and citrulline,
you can have a detailed discussion during the lunch.
So this is an NO-based, NO-directed mechanism which is very potent in nature, and it is homeostatic also. Because I just want to add here, we have this L-serine and palmitoyl coenzyme has a very strong phase 1 profile on the neuropathic condition. So basing on that, we are able to get the permission for the phase 2 clinical trial. And this is a nutraceutical. Even if it is taken in a high concentration, it will be utilized. It will be utilized in the protein amino acid pool. Yeah. So this is our mechanism. Yeah.
Thank you. So we may move on to next presentation by Dr. Om Prakash Singh, who is assistant professor at BHU. So he'll be talking about microplate whole blood interferon gamma release assay enables scalable detection of Leishmania specific cellular immunity. So over to you.
So, very good morning to everyone. And first of all, I would like to thank to the organizer for giving me the opportunity to discuss my work here. So, today I am going to discuss about our research work on microplate whole blood interferon gamma assay, enabling the scalable detection of Leishmania specific cellular immunity. So, visceral leishmaniasis is a parasitic disease caused by the Leishmania donovani and transmitted by the sandfly vectors. And before 2012, the visceral leishmaniasis was the India is the one of the most contributor of the VL disease all over the world. But nowadays, due to the effort of the VL elimination initiative, VL is on the verge of the elimination. VL is a disease of the poor. So it means that it occurs in a society in the people whose earning is less than $1. So you can imagine, if the people earning is less than $1 per day, how can these peoples are actually facing the challenges of the treatment and diagnosis? So this picture showing that the peoples are being treated at our hospital with the emphysema, and VL is not spread to the all over country. It is spread in only the 4 states: Uttar Pradesh, some part of the Uttar Pradesh, Bihar, Jharkhand, and West Bengal. So gold standard diagnosis is the splenic smear aspiration followed by the GIMSA staining, and this splenic aspiration is a very fatal sometimes. And the active VL patient are considered as the immunocompromised patient because their the PBMC are unresponsive to any of the antigens. However, in human subjects, we found the elevated level of the pro-inflammatory cytokines like the IFN gamma, TNF alpha, and other many cytokines. And we have also observed the elevated level of the mRNA in the blood of the active VL patient. So in 2012, we developed the whole blood assay just similar to the interferon gamma assay of the tuberculosis. So what we did, what we are doing in the whole blood, we collect the 3 ml of blood from the subject and divided into the 3 tubes, 1 ml each. And the first tube is considered as we do not add any antigen, and it is considered as the nil tube. And the second tube, we add the SLA, soluble Leishmania antigen. This is our experimental tube. And the third tube is the PHA tube, that is the positive control tube. And we incubate the whole blood for 24 hours. After 24 hours, we isolate the plasma from the blood samples and measure the cytokines in the plasma. So in our first experiment with the whole blood and PBMC, what we found that the surprising result that the whole blood of the active VL patient they are giving the IFN gamma response, whereas we did not get any response with the PBMC culture in 24 hours. After the 72 hours and 5 days, we did not get any response of the IFN gamma in either of the whole blood or PBMC. And the second thing is that we thought that because whole blood is giving the response, so whether it is the plasma is contributing some of the the association for the IFN gamma response or not. So we removed the plasma and watched the PBMC and blood, and then again added the healthy plasma to the blood and incubated the SLA. After replacing the plasma with the healthy blood, we still found the IFN gamma response, confirming that the whole blood is actually giving the response. And with this assay, we have conducted the series of the experiment on the 285 subject individual, and we confirmed that active VL patients are giving the IFN gamma response along with the IL-10 response. Now the question is, the whole blood is fine. It is the marker of the cell-mediated immunity, but we cannot use this whole blood assay for the field studies, because in the hospital we can take the 3 ml of the blood from the patient, but when we go to the field and we ask for the 3 ml of the blood from any of the subject, they usually deny to give the blood. So we thought that can we optimize this whole blood assay in the micro titer plate with the minimum amount of the blood. So for that, we actually divided our study experiment in 3 setups. In the first setup, we conducted the whole blood assay in the normal, similar way, where we collected the 3 ml of the blood. And now the second setup, we collected only the 600 ml of the blood and divided into the 3 microtiter vials, 200 ml of the each. And the third setup, we diluted the blood 50 to 50 and then plated on the culture plate with the 200 microliter of the blood and incubated with the 10 microgram per ml of the SLA antigen. And after that, we evaluated the IFN gamma response and found that even in the microtiter plate, the whole blood, assay microtiter plate is also responding and giving the IFN gamma response. And the diluted blood is also giving the response. But when we analyze the correlation between the whole blood with the 3 ml of blood with the 600 microliter of the whole blood, it was the correlation is the 80.5%, and whereas the 3 ml of the blood with the 100 microliter of the blood is the 79%. So to measure the agreement of the measurement of the 2 different methods, we did the Bland-Altman plot. And we found that with the 3 ml of the blood and also the 600 microliter of blood, the agreement is very high. So this suggests that the microtiter plate whole blood assay is also very sensitive and can be used at the field level. And this whole blood assay, microtiter plate whole blood assay, can be used as an alternative test of the Leishmania skin test. And because this is the field applicable test, we can combine this test with another test where we are collecting the blood, so we can evaluate the immune cells. Now the question comes in your mind that why we need this type of the test. So we need this type of the test because in the community, not all individuals who are infected develop the disease. Only the small fraction of the individuals are developing into the disease. So the question is what is others? It is because others have the cellular immunity, so they resolve their infection. So this is the cellular immunity. So we need to know the test by which we can actually investigate and monitor the cell immune person in the community. So I would like to thanks to the National Institute of Health and Bill and Melinda Gates Foundation for supporting for this study, and I would like to thanks to my participant for participating to the study. Thank you.
For timely completion of the presentation. Yeah. So maybe we can have the last presentation and if there are any questions, so that can be discussed during the lunch.
Yes.
So now the next presentation is by Dr. Manzoor. Maybe one question we can take.
Yes, ma'am.
I just wanted to ask, what antigens did you use? Protein antigens, or it's a combination of some other molecules?
So,
although I understand it may be a trade secret for you, but just tell us the macromolecule being used.
So this paper has been so the first paper we have published in 2012, where we have used the whole blood assay with the different antigens, like the parasitic antigens and the TB antigens and the parasite surface antigen, and we found that maximum sensitivity with the soluble Leishmania antigens, where we crushed the Leishmania parasite and isolated only the soluble proteins.
Okay, thank you.
Thank you. So we have next presentation, Dr. Manzoor Ahmed Mir, who is an associate professor at University of Kashmir. He'll be talking about the drug repurposing as a promising therapeutic strategy for receptor-negative breast cancers. Dr. Manzoor, I'm sure you'll restrict your talk to the 10 minutes. The speakers are requested to stay back for felicitation.
Last. This one. So good afternoon to all. I know this is lunchtime and the hunger pangs are already there. So I will be brief in my presentation and try to finish out within early as possible. So I will be talking about the drug repurposing as an approach of treatment of cancer by targeting the genome guardian mutant P53, which we all know that it is a transcription regulator, and we are targeting through it by different drugs which had already been approved for different purposes, but here we are using for that in breast cancer. So as we know that why this talk, because we had been talking about the women cancers in Kashmir. It has been on the rise since last 5-10 years, and these are the statistics which were presented at the Lok Sabha as well as in the assembly of the J&K government, where after that there has been a policy push last year, and the meeting for that was early in the Bharat Mandapam in the January month. After that, 2 calls had come up from the ICMR by this month. The last date for them is the 25th of June. That one is to reduce the survival of the cancers, especially the breast cancer in Kashmir. Another one is the prevention and the screening of the cancer in the Kashmir Valley, and it is a Kashmir J&K specific talk. So this tells us that this is a grave concern because the breast cancer is on the rise in the valley of J&K. So we had been discussing about it through different means that breast cancer is #1 cancer in the woman, and in terms of the mortality and in terms of the morbidity as well as in terms of the incidence. If you see here that every 3 minutes a woman is diagnosed with the breast cancer, and every 13 minutes a woman passes because of the breast cancer. So these statistics are very staggering. These are not the statistics, these are the tears rolling down from the families in whose these breast cancer patients are. So you see this is the statistics, and this statistics not tells about only this, it tells about us that the cancer is not killing just Indians, but the India too, because the cost associated with the cancer management is almost, it has been a scientific reports paper this year where they had estimated that it is almost $6 billion. And this is going to almost double to $14 billion in the next five years. So we have one more problem associated with the cancers here in this part of the world. One type of cancer is the triple negative breast cancer, which lacks the targeted therapies because there are no receptors available on the cells. And second thing is that this requires a chemotherapy and systematic therapies. So it limits the approach of treating triple negative breast cancer, which has highest incidence in Kashmir after the Nagpur. On an average, it is a 34.4%, where in Nagpur it is 45.3%. And TNBC cancers are most common in India, and India tops the list among all the countries across the globe in having the triple negative breast cancer incidence and mortality as well. On top of that, we know that the cancers in this part of the world are diagnosed in the late stage, because of which the survival chances are very less. So that is another problem. So when we talk about the survival as well as in case of the TNBC and non-TNBC patients, you will see that these two graphs, it is a clear distinction that in TNBC groups, the overall survival time is very less, as well as the relapse-free survival time is also very less as compared to the non-TNBC groups. So this brings us to the topic that why the chemotherapeutics? Why chemotherapy is required for TNBC treatments? Because it is the chief systemic treatment for TNBC despite multiple efforts to discover targetable therapeutic options. So in our lab, we know that these cancers can be targeted through multiple approaches. One is that antimetabolites we can use the mitotic spindle inhibitors like paclitaxel, which is the rationale of these therapies across the globe. Then we have alkylating agents, we have the other like enzymes, antimetabolites and all that. So we are also using the P53 modulator and topoisomerase inhibitors. So why drug repurposing or repositioning or reformulating or reprofiling? The same alternatives are there because you don't need regulatory approvals. The FDA approval is already there for a drug, and you just need to reposition it, reprofile it, or reformulate it, so that you need FDA post-market just oversee. You don't need regulatory authorities to go back to them and then guess. It reduces the time, it reduces the cost, and there are many other advantages to it as well, as is listed in the slide. So in our lab, what we are doing, we are using drug repurposing as well as combination therapy. If there is already approved drug for the cancer treatment, we are repurposing or repackaging that for the breast cancer, plus some drug which is showing promising effects. Here you can see that there is doxorubicin, adapalene, cryptolipine, zirambone, and many others listed in this case. So why this P53? Because we know that the P53 is mutated in most of the cancers, and in TNBC, almost 90% of the TNBC patients, P53 is modulated. So what here, this is a 393 amino acid long protein and a tetramer. So we know that this is a transcription regulator, and it is present in the nucleus, and then it is associated with the MDM2, and then it goes and binds to the DNA and activates that protein, which suppresses ultimately, because all the mutations, whatever are taking place, this is just guiding them to the Rapier mechanisms. When we went through the different databases, we found that P53 is mutated in 90% of the TNBC patients, as these different cell lines are the data from this TCG and other databases we have taken. Then we also see this, there are almost 7 hotspot mutations in this P53. When we next what we did, we compared the wild type with the mutant P53. So wild type P53 is usually associated with the MDMB2. It gets dissociated on this when it receives the stress signal in terms of that DNA damage or anything, any stress. It goes to the nucleus and binds it to the promoter region of that P53 gene, and then the translations and so on happens. It performs its function. But when it is mutated, you know that the MDMB2 is already dissociated from this. It goes, because it is mutated, it is not able to bind to the concerned promoter region of the DNA, and hence that it is not performing its regulatory function in case of the cancer cells. So what we did, we first doctored the wild type and the mutant P53, and we found that there are comparable doc scores, and both are working with the fine. And then we also found that these 7 DNA binding domain. There are 7 hotspot mutations which we had come across. What next we did, we took the DNA associated with that and the drug as well. So we docked the wild type P53 with the DNA, also with the mutant P53 with the DNA. Then we added our drug and docked also. We analyzed that CRP, that our drug cryptolipine, it is enhancing in the DNA binding ability of mutant P53 as the dock scores are increased as compared to the mutant one. We also validated these results by going to the MDA simulations and all that, computation analysis. Then we also validated this docking of mutant P53 protein by DNA chip assay or DNA pull down assay, where we are using the 2 concentrations of 50% of the IC50 as well as the 75% of the IC50. As you can see from the graphs, that there is significant enhancement when we are using the 3.4 micromolar concentrations in case of this cryptolipine drug. So we also checked the pro-apoptotic and anti-apoptotic molecules like BAX and PUMA,
Dr. Manzoor, you have 2 minutes.
molecules, then anti-apoptotic molecules, and we find that there is significant enhancement in the expression of pro-apoptotic molecules which is required, and there is significant inhibition in the these anti-apoptotic molecules. Then we also checked one of the approved drugs which was approved by the FDA, APR246. So when we did the docking studies of both these drugs, like our drug as cryptolipine and another drug as the APR246, we validated that results that our drug is working also fine with the mutant P53. So these are the results, Western blot results with the APR and the cryptolipine, which you can significantly see that there is enhancement in the expression of the P53 as well as that of the P21 in case of the treatment with our drug. So these are the RT-PCR results because of the paucity of the time. This is the cryptolipine drug, which these are all the functional assays. What we had done, like proliferation assay, MTT assay, and then SRB assay, then colonogenic assay and migration assay and so on. And we also did the cell cycle analysis and the apoptosis assays and found that cryptolipine is actually binding to the mutant p53 and causing its activation or remodulation so that it is able to perform its normal function. This is the mechanism what we proposed, and this is the overall study design, that it inhibits the proliferation, inhibits migration, colony formation abilities, it promotes apoptosis and cycle arrest as well. These are some of the publications which came out from this study of Dr. Hina Kuyum was performing these studies, and these are some of the papers which came out of this, almost 7 papers. We had filed also 2 patents in the initial stage with the US for one of the drugs, that is cryptolipine, and we are also modulating that cryptolipine. We are trying to find out its analogs as well. So this is the cryptolipine and its analog derivatives. So I will rush then another study which we did as repurposing. We are treating erdapiline with the GDC 0941, which is already approved drug. These are some of the functional results. So
thank you, Dr. Manzoor.
These are the results of this study we published from this. So another drug repurposing study we did with the GDC and doxorubicin. Then we are doing the paclitaxel and doxorubicin as well. So these are some of the books which we are publishing in addition to the to add to the knowledge domain.
Thank you. Thank you, Dr. Manzoor, for such an excellent work you are doing in the lab.
So last one slide I would like to add. So this we are organizing also this international conference on breast cancer. We had organized 2 in the last two years, in 2024 and 2025. This is the 2025, and the third international.
So the people have reason to come back in September.
In September, because the temperatures will be cooling down up to that date. So anybody who wants to come to that conference, most welcome. Thank you.
Thank you so much. Okay. So maybe before I ask, okay.
Yes. Okay.
Wait. I too have a question.
So in vivo we had not gone. We had just tried till the computational ways, in silico wise, as well as the in vitro wise with the functional assays. One of the students is working as in vivo models. We have got the ethical clearances for that. He will be doing that.
So Dr. Manzoor, is it like you are talking about the repurposing of a drug? Is it like, dose escalation or de-escalation using the compounds? Are they specific to TNBCs or like it is general? And the other question is, like you said, P53, like I understand you showed 90% involvement of P53 mutant. Is the P53 cause or a consequence in TNBC? Yeah, we'll discuss during the lunch.
Thank you.
So
we'll discuss
during lunch. So he can give my questions during the lunch.
So before we go for lunch, I would request our chairs to felicitate our speakers. I would request Dr. Shaukat Hussain to felicitate our first speaker, Dr. Anwar Alam.
May I now request Dr. Khalid Hussain to please facilitate our second speaker, Dr. Monica Sharma.
Now I request Dr. Shaukat Hussain to felicitate Dr. Mithilesh Kumar Singh.
May I request Dr. Khalid Hussain Bhatt to please felicitate Dr. Shubhankar Kumar Singh.
Now I request Dr. Shaukat Hussain to felicitate Dr. Srikant Sadhu.
May I request Dr. Khalid to felicitate Professor Hidesh Prakash.
Now may I request Dr. Shaukat Hussain to felicitate Dr. Om Prakash Singh.
May I request Dr. Khalid Hussain to felicitate Dr. Manzoor Ahmed Mir.
Now at last may I now request Dr. Khalid Hussain to felicitate Dr. Shaukat as he is leaving for Delhi.
We will now adjourn for lunch break. Kindly reassemble. He'll continue. We'll reassemble exactly after 1 hour. So have a nice lunch break. Thank you.
He'll be speaking on gut-residing bacteria of VL non-transmitting sandfly as a novel therapeutic and preventive invention against drug-resistant Leishmania donovani.
Okay. Hello. So slide is working? Okay. I have a laser there. Okay. Anyway, thank you very much. Thanks to the organizing committee for providing me the scope to deliver or to discuss the work which we are doing at my university, West Bengal State University. Basically, I'm working on something different type of work. It's not immunology, it's not molecular biology, it's not a vector biology. Little bit about on vector molecular biology. So what's the vector molecular biology? Vector molecular biology is the combination of little bit amount of insect biodynamics, handful of molecular biology, and good amount of immunology. So what I will discuss today with you, the identification and characterization of guard bacteria from disease non-transmitting sandfly vectors against the drug-resistant Leishmania donovani. Before to go, we first need to understand what's behind the vector bites. Only anticoagulant? no, this is not only anticoagulant. There are several salivary gland proteins and several gut bacteria. Those are basically ingested or inoculated during each bite of either disease transmitting or disease non-transmitting vectors. So this is about the system, how these vectors basically introduce the salivary proteins and the gut bacteria to the hosts in exchange of their blood meal. They have a particular specific pump system here, which is known as Siberial pump, and which has lever that acts as a hypodermic syringe, that acts like a hypodermic syringe, and it evacuates the air, it evacuates the salivary gland proteins and the gut bacteria into the system of host system in exchange of blood. So these are the basic CT scan structure of the blue one is the Siberial pump. And one thing you must remember that insect mouth purse, they have a bizarre pattern of mouth purse. It's an excellent evolutionary viewpoint. If you check the insect mouth purse, the sandflies, they have a strong maxilla, and both the ticks and sandflies, they have a strong maxilla, and they can create a well into your skin, so that a well filled up with the host bloods they can create, and thus they take the blood, thus they can take the blood through their proboscis. Whereas in case of mosquitoes, what they do, they insert their proboscis up to the blood vessel level, thus introduce the salivary proteins and the gut microbiota, and takes the blood from it. So what is the hypothesis about my work? In tropical countries, in India, in Brazil, both the disease transmitting and disease non-transmitting vectors, they coexist. In case of visceral leishmaniasis, Phlebotomus argentipes, this is the disease transmitting sandfly, whereas Phlebotomus pepatesi and the Sargentomia species, they are VL non-transmitting parasites, VL non-transmitting sandflies, they coexist. And also the several type of mosquitoes. So, in tropical countries, one cannot ignore the opportunity or chances of getting bitten by both VL transmitting and VL non-transmitting sandflies. So we know that Argentipus, Phlebotomus argentipus, they can carry and transmit the Leishmania donovani, the causative agent of visceral leishmaniasis. However, the Sargentomia species and the Phlebotomus pepatesi, they cannot carry the Leishmania donovani. However, they can carry other leishmanial species which are not causing the visceral leishmaniasis. But as both Sargentomia and pepatesi, during their bite, they can introduce the salivary proteins and their gut microbiota. So, what is entirely missing? What is the influence of these proteins and gut microbiota on the pathogenicity of VL? So the work started after coming back from NIH through ICMR faculty fellowship. Before that, we have been working on the Leishmania drug development and the Leishmania immunology. And after coming back to 2017, we have started the vector molecular biology. So when we started, we found South Chopra Canning is nearby field that is of my university, and we observed a few Argentipus or abundance of few Argentipus and pepatesi. However, huge number of Phlebotomus, huge number of Sargentomia species were there in Canning, which is supposed to be the Leishmania VL endemic region up to 2012. However, very few VL cases we observed, or there are reports from the West Bengal government. You can observe after very few VL cases after 2012. So at first we found two different type of Sargentomia there. We just named these OFs 1 and OFs 2. Morphologically, during the morphological identification, we found they have two different type of sidereal which is called sidereal teeth, and these are OFs 1 and OFs 2. Interestingly, one morph who has small number of teeth that is found to be take blood from both the reptilian and the human species. The significant, which are the significant? Basically, Phlebotomus argenteus is basically zoophilic. It prefers warm-blooded animal, whereas Phlebotomus pepatus is anthropophilic. So, Sargentomia species all over the world, they are zoophilic, but they prefer cold-blooded animal. However, as we found a specific type of Sargentomia species we have identified as a human probing Sargentomia sandflies, disease non-transmitting sandflies, we continued our study along the West Bengal. So, this is the South Chobbish Pargana. This is the South Chobbish Pargana. Initially, we have started this one, and then went to Malda, and then went to Darjeeling district. The point to be noted that South Chobbish Pargana endemic till 2012, but not now. However, Malda is highly endemic, and Darjeeling district, Fashidewa region, they are endemic, but not as good as, the Malda region. So, we started the observation in this region, and then we found there are several types of Sargentomia which are prevailing in West Bengal are Sargentomia babu, Sargentomia shorty, and Sargentomia punjabensis. So up to this thing is morphological, nothing immunology in it. However, as we observed, a specific type of Sargentomia babu, it can take bloods from the humans. So we started the analysis of blood means, and we found that this is the lizard CY based on our PCR, single fly. We have isolated the mRNA from the single fly, and then we have done the PCR. This is the human ALU specific, and we observed that in South Chobbish Pargana district, which is VL non-endemic at present, there are probability of biting this human probing Sargentomia species higher than the Malda and the Darjeeling districts where still visceral leishmaniasis is endemic. So the first part of the work is we have identified a human probing Sargentomia. Very few are reported from all over the world. And as
Professor Pal, we're short of time. Can you please wind it up?
Short of time?
Yeah.
Okay.
Our time is actually up.
Kitna time hai?
1 second. 5 seconds.
Oh, so we have identified what we did. We isolated the gut bacteria and tried to identify which are the anti-leishmanial effect. Okay, just 2 minutes. So we found some bacteria which are anti-leishmanial effect, and we found among this SB1, SB12, SB17, and SB22. These are the 4 species, among which SB12 is the most important one. Next one is the identification of the root, and this is the toxicity we have identified from in the bone marrow, which is these bacteria are non-toxic. And interestingly, what we found that these bacteria couldn't induce so much amount of IL-12. However, another cytokine which is IL-23, and IL-23 and in combination of IL-1 beta, these basically induce more amount of IL-17 rather than the IFN gamma. So next we have identified, the expression of IL-17 from both CD8 and CD417 positive cells, and we also found the ratio of induction of IL-17 is more than the IFN gamma. So another most point, as it has a prophylactic effect, we identified both the ROS positive and TNF-alpha positive monocytes are higher during the prophylactic approach of this bacteria. And based on the data, ICMR interestingly submitted a patent on our observation, and I'm thankful to ICMR. They have spent all expenses, and it's a collaborative patent with ICMR and our university, West Bengal State University. So the take-home message is, to the best of our knowledge, this is the first report for the development of a prevention and treatment of drug-resistant Leishmania, Donovan infection by the Hendrixia's porothermodurans. We have already, identified this bacteria with the next gen whole genome sequencing, etc. And a MS has been already submitted. So these are my few previous articles, and these are my lab members. These work based on Shumon, Kormokar, and Kamalika Roy and Pritam Mondal. And
so thank you for an interesting talk.
So thank you very much.
So
this is my
due to paucity of time, we will not have time for any questions. So can we move to the next speaker, please?
Thank you, Bro. Institute of Medical Sciences, New Delhi. He'll be speaking on Treg-mediated immune suppression in visceral leishmaniasis, linking parasite burden to host immune dysregulation and therapeutic restoration. Please stick to your time, sir. Stick to your time. Yeah. 10 minutes?
Well, more than sufficient.
Thank you.
So, good afternoon to all, and thank you for the organizer for giving me this platform to talk. And before going to start my talk, it's better to give a disclaimer for my study. So whatever the data I will be talking in this today talk, those all data we had generated few years ago, couple of years ago. So title of my talk is Treg-mediated immune suppression in visceral leishmaniasis leads to the parasitic burdens. So, as we know, visceral leishmaniasis or leishmaniasis is a neglected tropical disease, and this disease has 3 different forms. Visceral leishmaniasis is one of the deadliest forms, and which is fatal if not get treated. Other 2 are cutaneous leishmaniasis and mucocutaneous leishmaniasis. And the associated factors for this disease are poverty, malnutrition, population displacement, climate change, and poor hygiene. So there are 8 countries all over the world, those are contributing the visceral leishmaniasis, and in that India is among of them. So this is the world map showing the density of visceral leishmaniasis spread all over the world, and this is the data from 2022. And however, in India, the visceral leishmaniasis cases significantly decreased due to the better implementation of eradication program by government of India, but still the prevalence is very high, and that comes under 500 to 1000, means deep concern. So you can see in India there are only 4 states where this disease is endemic, and those states are already mentioned here. But the remarkable things I had put it in this table, that after 2020, the disease has been decreased drastically. In 2020, it was 2048, and now in 2023, somewhere in 520 to 595, and other reports are still awaited. So this disease has gone decreased with effective implementations of Government of India policies. So this is the transmission pattern of visceral leishmaniasis parasites. This parasite is digenetic parasite, so it requires 2 different hosts. Primary host is human, however the secondary host is sandfly. So whenever any sandfly bites any human individuals, they transmit the promiscuous form of parasites into the human, and that promiscuous form converts into amesticate forms and comes inside the macrophage. And inside the macrophage, they continuously grow or increase their number, and after that they brush the infected macrophage and release the parasites, and those released parasites again infect the other macrophage. At the same time, if the scent fly bites that infected individuals, they again took the parasites, and those parasites comes inside the scent fly and convert their form into promiscuous to promiscuous forms, and those parasites are ready for fresh infection. So, since this disease is a intracellular pathogenic disease, so the Th1 types of immunity is essential for protections of individuals. And so whenever any parasites or intracellular parasites comes inside the host, they taken up by the antigen presenting cells, and those antigen presenting cells process and present those antigens to naive T cells. And in presence of antigens, those naive cells are activated and produce the pro-inflammatory cytokines, predominantly Th1, predominantly IFN gamma and TNF alpha. Those IFN gamma and TNF alpha acts on macrophages, and they activate the macrophages to produce the reactive oxygen species and nitrogen species. These reactive oxygen and nitrogen species are highly reactive in nature, so it try to kill the infected macrophage along with the parasites. And through this, the host will be protective. But here, the parasite modulate the host immune system in their favor so that they can continuously grow inside the host by decreasing the concentration or frequency of pro-inflammatory cytokines, specific cell or their soluble cytokines, or modulating the suppressive immunity. So this talk is completely based on human-based study, and my talk is completely based on the Treg-mediated suppression and how these Tregs are influenced by the parasitic load in the visceral leishmaniasis patient. So these are the different mechanisms through which the Treg cells suppresses the T effector cells. First of most is the ATP is known as the pro-inflammatory conditions, and Treg cells are predominantly expressed in CD39 and 73. These are the ectopeptidases, and these ectopeptidases convert pro-inflammatory ATP into adenosine, which is suppressive in nature. And presence of adenosine decrease the effector functions, and that leads to the bacterial favors. In second hand, the Treg cells produces the IL-10 and TGF-beta. Those IL-10 and TGF-beta are suppressive in nature. Again, they suppress the effector cytokines producing cells. Next mechanism for Treg-mediated suppression is by generation of cytolytic molecules like perforin and granzyme. Those perforin and produced perforin and granzymes are cytolytic in activity, and they kill the effector cells as well as the other immune cells also. And Treg cells are predominantly known for the high expressing of CD25 molecules. CD25 is known as the IL-2 receptor, and IL-2 is essential for clonal expansion of T cells. So in deprive of IL-2 in the milieu, the T cells are not able to clonally expand and increase their effector function, effector number. So this study is completely based on 26 visceral leishmaniasis patients. These patients came in the clinic with these clinical features, and these patients were first screened for RK39 positive and further validated for the visceral leishmaniasis, BL LD bodies demonstration in bone marrow. So, and after that we scored them +1, +2, +3 based on the as per the WHO guidelines. And these are the representative plots for the LD bodies demonstration in GIMSA staining. We had divided these group of patients into two groups, high parasitic load groups and low parasitic load groups. The patient, the parasitic burden was very high, we scored them the +3 and the +3 and +2, and the patient whose the parasitic load was low, they were graded as +1. So there was two groups, one group low parasitic load groups where the +1, and high parasitic load groups 2 and more above than 2. So this is the first result, enrichment of Treg cells in bone marrow. For that we isolated the MNC from bone marrow aspirates, and then stained the isolated MNC for Treg specific markers CD4, 25 and FOXP3. And CD425 cells were demarcated as 25 dim and 25 high populations. And further, we evaluated the CD25 dim and CD25 high for FOXP3 expression. And we observed CD25 high population were predominantly expressing FOXP3. So, and that population we got majorly in high parasitic load group compared to the low parasitic load groups. And, sorry. So, in high parasitic load groups, again, we observed the high level of CD425 population compared to the low parasitic load groups. Again, the FOXP frequency was again significantly high. And further, we evaluated the T effectors of frequency. T effector cells we enumerated as CD4, CD25 positive, FOXP3 negative. And that frequency was significantly low in high parasitic load group compared to the low parasitic group. Further, we did the correlation study in that the FOXP3 positive cells were correlated with the CD425 populations, and that was positively correlated. Furthermore, we correlated the FOXP3 frequency with the parasitic load, and that was again positively correlated. So with this study, we conclude that the Treg cells are significantly enriched in bone marrow of BL patient and increase the parasitic load, and parasitic burden is associated with enhanced immune regulation, suggesting a role for Treg in parasite persistent and T cell suppressions. So next, we were interested to see what exactly the impact of these Treg cells in IL-10 productions. And we observed the CD425
Dr. Kumar, we're short of time. Can you please finish?
Just 1 minute. So CD425 high population were expressing high IL-10, and that was significantly high in high parasitic load groups compared to the low parasitic load groups, and that was corroborated with the soluble level of IL-10. So the parasitic load is associated with increased frequency of IL-10 producing Treg cells in bone marrow of BL patient, and enhanced IL-10 mediated immune regulation may contribute to T cell suppressions and parasite persistence and visceral dismiss. Next, we observed what exactly the impact on the pro-inflammatory cytokines. And in that, we had taken the two cytokines, specifically IFN gamma and TS17, and that was significantly high in low parasitic load groups compared to the high parasitic load group, and that was negatively associated with the Treg frequency. High parasitic load is associated with diminished IFN gamma and IL-17 producing frequency, and suppression of TH1, TS17 response may contribute to the inefficacy parasite clearance, persistence, infection, and visceral dismisses. Next, we evaluated the soluble level of cytokines in which IL-6, TGF-beta, and IL-17 mainly. And we observed the IL-6 and TGF-beta were significantly high in high parasitic load groups, and IL-17 was significantly high in low parasitic load groups. And their ratio was significantly high in high parasitic load groups. So this is the rescue. This is just last slides, and this is the for the rescue. For rescue of T effector functions, we block the IL-10 and TGF-beta alone and in combination. With alone and in combination, we observed the rescue of pro-inflammatory cytokines producing cells, specifically IFN gamma and TS17 cells takes place. So these are the summary. Visceral dismiss chronic infections, and the Treg cells are directly proportional to the parasitic load groups in bone marrow. And the degree of T cell dysfunction is directly associated with diverse severity, suggesting that the parasitic burden is a key determinant in immune potency. Thank you.
Thank you so much. So owing to the paucity of time, we will not be able to take any questions. So can we move to the felicitations, please?
Thank you so much, speakers. Thank you so much, worthy chair, Dr. Khalid. May I now request Dr. Khalid to felicitate our speakers. First up, may I request Professor Chiranjeev Pal to come to the dais, please. May I request Dr. Prabeen Kumar to come to the dais. May I request our worthy chair, Dr. Khalid Hussain, to felicitate our speaker, Dr. Prabeen Kumar. May I request Dr. Khalid Hussain to grace the chair, please. for 2 more talks. We have postdoc presentations. First up, we have Dr. Wasim Ali from Sharda University. He will be speaking on exploiting thymine biosynthesis as a metabolic vulnerability for multi-target inhibition of Mycobacterium tuberculosis. A quick update, there is a poster session going on upstairs from posters 1 to 80. If any of you belongs to that category, 1 to 80 posters, please go upstairs. Dr. Wasim, please go ahead.
Good afternoon, everyone. I'm Dr. Wasim from Sharda University. First, I would like to thank the organizer to give me the opportunity to present my work here today. In recent time, we all have experienced the COVID-19 pandemic, but since last 3 decades, we all are living in the pandemic of Mycobacterium tuberculosis. WHO declared MTB a global pandemic in 1993, and it is a global pandemic since then. According to WHO report, in last year only, more than 10 million people have been affected by Mycobacterium tuberculosis, and the raising of drug-resistant type of TB make it different to treat Mycobacterium tuberculosis. And the urgency of identify a new novel target are there, and to identify the novel target in Mycobacterium tuberculosis, we have focused on thymine biosynthesis of Mycobacterium tuberculosis, which is essential for the growth of Mycobacterium tuberculosis. And importantly, the human do not synthesize thymine biosynthesis that make this pathway a pathogen-specific pathway. In this pathway, there are 4 different proteins which are essential and shares no homology with the human protein, which is important to avoid the non-specific inhibition of human protein. We want to kill Mycobacterium tuberculosis, but we do not want to affect the human protein. For this study, we have taken a polypharmacological approach, and we have targeted all these 4 proteins, and we try to identify a drug which can go and inhibit all 4 proteins, which will ultimately reduce the chances of emergence of resistance in Mycobacterium tuberculosis. All these targets are absent in human. Then we are taking a drug repurposing approach. In conventional drug discovery, it will take about 15 years. And in drug discovery, you can reduce the time, labor, and the invested money because the drug are already been approved by FDA, and you do not need to perform all the cytotoxicity experiments. So we hypothesize that if we are able to inhibit this important pathway of Mycobacterium tuberculosis, we will able to inhibit the growth of Mycobacterium tuberculosis, and we can have one more drug for Mycobacterium tuberculosis treatment, and we can also reduce the time. For drug repurposing, you can have, the prime example of drug repurposing is metformin drug, which was approved for diabetes, and now we are using that in treatment of Mycobacterium tuberculosis as well. And here in this study, we are also trying to replicate that scenario. Now, we have more than 3,000 FDA-approved drugs, and we cannot start working on all the drugs at once. So we have performed molecular docking to select a drug which can go and bind with all 4 drugs, and we are able to find a drug which can bind with all 4 protein with good binding affinity. I'm not showing the name of the drug here, and I identified that drug as drug X because the data has not been yet published. And after that, in protein docking, there is only protein and the drug. To check the stability of that complex, we have performed molecular dynamic simulation, which mimic the physiological condition of human, and we found out that our drug with all 4 targets has been stable, and we proceed for in vitro to check whether our in silico hypothesis is actually translating into the wet lab work. We first investigate the MIC of that selected drug, and the MIC of that selected drug is 3.12 microgram per ml. In the same experiment, the ethambutol, which is the first line drug for Mycobacterium tuberculosis, it is showing 6.25 microgram per ml. So our drug is showing better MIC and killing ability than the first line drug. Then we have performed the growth kinetic to check the effect of a drug on the survival and progress of MTB. And then after 7 days, we have plated the bacteria for CFU count to check that reduction in the OD is equivalent to reduction in the viable cell of MTB or not, because MTB also produce a lot of clumps and it can enhance your OD. Then, as we all know that The treatment of Mycobacterium tuberculosis is a combinational therapy. So we have checked the efficacy of our drug with ethambutol, and when we are giving our drug only at lower of MIC value, it is showing around 25 to 30 microgram, 30% killing. But when we are using it in combination with ethambutol, it is killing approximately 80% of H37RV, which indicates the synergetic effect of our drug with the first line drug ethambutol. Then we want to kill the Mycobacterium H37RV, but we don't want to harm the THP cells. We do not want to harm the macrophage of human. So first we have checked the cytotoxicity of our drug on THP1 cells, and the drug is not cytotoxic. Even at the 10x of the concentration, we have used the LMR blue dye on that.
But Ali, wait until last 2 minutes so that you try to finish it up so that we can have some time for the question from the audience.
Then the macrophage, the MTB is usually present inside the macrophage. To check intercellular clearance by our drug, we have performed the ex vivo experiment, and we can see that our drug is able to clear the MTB more than 95%, which is better than the isoniazid also, which is the first line drug. Then we have gone to in vivo experiment. We have infected the mice with the vehicle control, our drug, isoniazid, and our drug plus isoniazid. And this is the last way. We can see there is an improvement in the pathology of the lungs. First is the healthy control, infected control, and we can see that only isoniazid, only our drug, and isoniazid plus our drug is showing the better morphology of our drug. And also the most important thing from this study is that isoniazid is able to kill 97% or 89% in 2 weeks, but our drug alone is able to kill 97-98% of the bacteria from the lungs. And the main objective of this study was to reduce the timing of the therapy of MTB, which is normally 6 months, and it can lead to a year as well. And in this study, we are trying to lower down that time, and our result from in vivo experiment, it is showing that we can reduce the time from 4 weeks to 2 weeks. This is the conclusion that now we have one drug which can also be used as the adjuvant in the combination therapy of Mycobacterium tuberculosis. The further study will be checking the immunoadaptation and the relapse and whether this drug is able to conquer that or not. Then I would like to thank my team. Professor Ehtasham Hasnain, Professor Nasreen, Professor Anwar Alam, and
thank you, Dr. Ali.
Thank you very much.
Can we have any questions, please, from the audience? Okay, I think we are short of time also. So, can we move to the next postdoc presentation from Dr. Mohammad Musa Mubarak? He's from Department of Clinical Biochemistry at SKIMS. Do we have him here? Please. So can you please stick to your time? And just to remind you that you have only 7 minutes. We don't have a 10-minute talk, okay?
Okay,
thank you so much.
Okay. How do I get the presentation? I have mailed you this. It's here.
TB presence with light microscopy using ZN staining, and TB presence with TB PCR. Now, in order to first confirm my in vitro model, I confirmed that my cell line was indeed U87Mg cells, astrostick cells, with STR profiling. Then this is the infection studies here. The graph here shows on my left-hand side, it shows the number of MTB internalized by these astrostick cells. So as it is clear that these astrostick cells internalize this Mycobacterium tuberculosis bacilli in dose and time dependent manner. The downside of my left-hand side slide here confirms the presence of MTB as pink bacilli. Their PCR bands confirm that these cells, these bacilli obtained from the lysates were indeed Mycobacterium tuberculosis. So this slide, I conclude that these astrostick cells, they do not just showcase their support for the cells in the nervous system, but in fact take part in defending the central nervous system by internalizing these MTB bacilli. Now, next, I studied what was happening to the cell viability upon MTB infection. Here again, as it is clear that the viability of astrostick cells was getting decreased with increase in load and incubation time. Next, in this fluorescent microscopy study, I confirm that this change in the astrostick cells upon MTB infection is due to mitochondrial membrane potential loss. Here, we observe that these astrostick cells, they don't just die when faced with MTB challenge, but indeed respond to this MTB infection by releasing nitrous oxidus species. And again, here in this study, I show that these cells bring on the immune response by secreting ROS. Now, the main key component here is the release of this matrix metalloproteinase 9, which is a ECM modulator, and it is this entity that further activates the cascade of immune response to MTB challenge in the CNS. Here in this slide, the MMP9 presence is confirmed by the zygomorphic gels. Here again, the same pattern is followed, that with increase in bacterial load, increase in multiplicity of infection, there is increased release of MMP9. There again, with increase in the incubation time, there is an increased release of MMP9 by these astrostick cells. Here, these slides show that besides MMP9 and other basic immune responses, astrocytes also bring forward the release of these inflammatory markers in a dose-dependent manner to the incoming MTB challenge. So here are some conclusions. Next, we observed that the key thing connecting MTB infection in the CNS, the astrocytes, and the entry of MTB into the CNS was this MMP9. So we devised our in vivo studies wherein we infected Balbc mice and gave two treatments, cyclopamine treatment and puromorphamine treatment. Cyclopamine treatment, it is an MMP9 antagonist, SHH antagonist, which means that it will increase the secretion of MMP9, whereas puromorphamine is SHH, it increases the secretion of SHH, thereby preventing MMP9 secretion. Then we did this mice sacrifice and these assays. Now, from the brain homogenates, in our first group, which was just given sham treatment, we see there was no MTB bacilli found from the brain homogenates. In the second group, where MTB infection was given and then there was no treatment given, we see there is some MTB presence. In the puromorphamine treatment, we see there is attenuated decreased presence of MTB. And in cyclopamine treatment, we see that there is heightened increase of MMP9 presence in the brain, thereby confirming that MMP9 modulates, changes the shape of the ECM, thereby facilitates MTB entry into the brain. This we confirmed with our histopathological studies. Here, uninfected group, infected group, puromorphamine treated group, and the cyclopamine treated group are totally different. This we again confirmed with LCA treatment, with LCA studies, IgG studies, and GFAP studies. And here, GFAP, this IHC marker, confirms that there is massive architectural changes upon MTB of entry into the CNS, and this is a classic marker of astrocytes. This we here by ELISA studies proved that this is by MMP9, and Western blotting also confirmed that.
and this is my conclusion for this study, that indeed it is MMP9 that is responsible for changing brain architecture, thereby facilitating the entry of MTB into the CNS. Here I would like to acknowledge Dr. Zohra Ahmed Pari, my previous PhD guide, Director SKIMS, for giving me space in SKIMS as an NRF and PDF, DST Inspire Fellowship for conducting my PhD work, and ANRF National Postdoc Fellowship for continuing this work of mine in patient samples at Sher-e-Kashmir Institute of Medical Sciences. Thank you so much.
Thank you so much, Dr. Musa.
Oh, that's it. Thank you.
May I request Professor Pawan Sharma on the dais, please? May I request you to kindly felicitate our worthy chair, Dr. Khalid Hussain. Thank you so much, sir. Thank you so much, Dr. Khalid. We now disperse for the poster session, which is being held upstairs. Students, they come all the way from different parts of the country just to get our expertise, just to get some sort of guidance and acknowledgement for their work. If I could request you to kindly go upstairs and see what and all work they are doing and how and all we can, guide them for a better, study ahead. Thank you so much. We gather here again at sharp 5 p.m. with plenary talks and IIS award talks. Thank you so much. Dr. Sarat Dalai from the North Zone. Please welcome Professor Oma Kanga from All India Institute of Medical Sciences, New Delhi, and from the South Zone, Dr. V. Aravindan from the University of Madras. I request all the chairpersons to kindly take their chairs. It's my proud privilege to introduce the recipient of this year's IIS Mid-Career Scientist Award, Professor Deepyaman Ganguly, professor at Ashoka University. Professor Ganguly is a physician-scientist whose research has illuminated the biology of plasmacytoid dendritic cells, type 1 interferon signaling, and autoreactive inflammation. His work spans fundamental immunology and translational research, including the development of toll-like receptor antagonists and immune-based therapeutics. He has pioneered studies on piezo-1 mediated mechanosensing in human T cells, a truly frontier contribution. A recipient of the Shanti Swarup Bhatnagar Prize, the National Bioscience Award, and the Swarna Jyoti Fellowship, he is an elected fellow of the Indian Academy of Sciences, the National Academy of Medical Sciences, and the Royal Society of Biology. Ladies and gentlemen, please join me in warmly congratulating and welcoming Professor Deepyaman Ganguly. The floor is all yours, sir.
A very good evening to all of you. It's a special moment. I don't know how many of you know. Myself and Amit also had been an editor as a student in Indian Immunology Society, and that was 2005. And this is 2026, right? So 21 years after your own society recognizes your contribution, I think it's a great moment. Thanks to the IIS, all IIS members, especially the executive committee and all the chairs who are there. I'll talk about, so how do I, So I decided that I'll give you a brief glimpse of a journey that we have been continuing over the last 1 decade. And before I get into that, I would acknowledge the contributions of all the students who have been through my lab, because the reason I stand here because they work, right? So I'm just actually representing my students. And I have been extremely fortunate in having extremely motivated students. They have gone to places, and I still have 2 students in the new place that I'm now in, which is Ashoka University. I spent almost a decade, more than a decade, in Indian Institute of Chemical Biology since 2013, and recently I have joined Ashoka University. I was also very fortunate to work with brilliant collaborators, extremely well-meaning, generous collaborators who have been working with our group over the last 10 years or around 12 years. Now, coming back to our journey, I started this journey with plasmacytoid dendritic cells almost 22 years ago. And so 2 decades we have been working on this. First decade was spent in learning the craft from the masters, where I was actually working with people. I was, again, very fortunate to work with people who were pioneers in the field. And the next decade, I tried to do in my small way to contribute to the understanding of these really enigmatic cells. Now, these are dendritic cells. There is no need for introducing dendritic cells to this audience. But PDCs, we all know about dendritic cells as the professional antigen presenting cells. But PDCs are little different. They don't even have the dendritic morphology. They really look like lymphocytes. And people were, for a long time, people were really confused whether they are, what kind of cells they are, whether they are lymphocytes or not. And the major function of these cells, and through which it was identified also long back by Giorgio Trinchieri. You know Giorgio Trinchieri for his discovery of IL-12, but it was Giorgio who first saw there are some HLA-DR positive cells that are producing a whole lot of type 1 interferons when you are throwing in bacteria and viruses in the blood, human blood. And that's how these immune cells were first described. And interestingly, Giorgio was accompanied by someone called Dr. Shantu Bandopadhyay, and I was extremely fortunate to do a PhD with Shantu. And so PDCs are professional interferon producers. Why they are then called dendritic cells? Because they share the ontogenic origin, so hematopoietic origin. They all come from the common DC precursor, although over the last 5 years we have some understanding that some of the PDCs can actually come from lymphoid progenitors. But they don't really have this dendritic morphology. What they have is a very intricate Golgi network, because these are really secretory cells. And in response to the stimuli I'll talk about, through which it starts producing interferon, when they get this stimuli, they can actually produce nanograms of interferon if you're taking like 10,000 cells, which is huge for any immune cell in terms of producing such a high amount of protein in a very short period of time. And that's why perhaps this extremely intricate secretory network, extensive Golgi network, and they look like, under microscope, plasma cells, because that's the other secretory cells which has a very intricate Golgi network. And that's why the name came plasmacytoid disease. And we have been, we know now that the stimuli that majorly drive these cells to produce type 1 interferons are toll-like receptor activation. These toll-like receptors are not on the surface. They are inside the endosomal compartment, and there is a reason for that, because these toll-like receptors are, they recognize nucleic acid ligands. And if they were on the surface, the cells will be always activated, because our body is always full of nucleic acids which are circulating, and they are released by all the dying cells in the body. So naturally, these receptors are present within the endosome, so whatever is being taken up by the cell inside that endosomal compartment, then it can activate the toll-like receptors, and they produce type 1 interferons. Now, these nucleic acids can come from pathogens and also come from self-origin genome, like self genome, and when there are host cells dying and nucleic acids are released, they can bind to certain proteins, certain danger signals in the body that can take these self-nucleic acids into the endosomal compartment, and then they can start driving PDCs crazy, and they will start producing interferon. And naturally, with this discovery, started PDC's journey in the autoimmune diseases, because naturally it was quite clear that they have the potential to drive a whole lot of autoreactive inflammation, and it was first shown in the context of PDC, in the psoriasis, and then also in the context of SLE. So our lab has been looking at role of PDCs in chronic autoreactive inflammatory diseases, not only in autoimmune diseases. We started with, as I already mentioned, the original discoveries in psoriasis and SLE, which was done while I was a PhD student by Michel Gillier and Yongjun Liu. By the way, Yongjun Liu was he's credited to be the actual discoverer of PDCs because he described them as dendritic cells. Giorgio did not do that. And we have been looking into this. We started our journey with SLE, which continues with psoriasis through metaflammation, and slowly we are getting into inflammaging and looking at a more holistic view of how autoreactive inflammation can happen as in an urbanized world. However, big fraction of my PhD students always had been extremely interested in the cell biology of PDCs. I'll briefly talk about that journey today. So all these chronic autoreactive inflammatory contexts, you need big patient cohorts. Because of my background, I have been working with a lot of clinicians, and so those are mostly driven by a team. But in case of the cell biological programs, usually individual PhD students took up the responsibility and produced those data, discovered interesting regulators, molecular regulators in PDCs. Now, why specific molecular regulators in PDCs are so important? As I already mentioned, the TLRs are inside the endosomal compartment, and there have been seminal work by Greg Burton and Jonathan Kagan, who showed that TLR TLR9, for example, which recognizes DNA, or TLR7, which recognizes RNA. So TLR9 had been shown to be present on the cell surface, and it was a really enigmatic finding, because then the whole reason for having these TLRs inside the endosomal compartment goes away, and then Greg Burton showed that, they do come onto the surface, but they are inactive. They are proto-TLRs. They can become only active when they come inside the endosomal compartment, because within the endosomal acidic pH, the cathepsins will actually cleave off a part of these TLRs, and then they become active. And that's how the cell maintains sort of sterility on the surface, but TLR or pattern recognition function within the endosomal compartment. And then you are also thinking of all these nucleic acids which are being taken up by different pathways, click-GIG pathway, macropinocytosis, etc., which will land up in the endosomal compartment, and naturally the endolysosomal trafficking have to be tightly regulated in PDCs. And it has been a long-standing interest of us why PDCs are so proficient in measuring how much response it will actually create in response to a TLR ligand, and also I'll come to that later. And there are fine-tuning in terms of where exactly in the endolysosomal pathway TLR is getting activated, and that also defines how PDCs function. So our students have been looking at different parts of the regulators. For example, there have been studies on extrinsic regulation where like biochemical mediators from outside the cell, exogenous from other cells, can actually influence PDC function. For example, Deblina Rai-Chaudhury first looked at that lactate, which is present in the chronic inflammatory context as well as in the tumor microenvironment, actually can drive PDC dysfunction through GPR receptor, which is a lactate receptor, which is again present on PDCs, GPR81. And that also drives PDCs to play a more regulatory role in terms of driving T regulatory cell expansion inside the tumor. Then Wendrila Rahman looked at a very interesting pathway which we never thought will be involved in PDC biology, and we came up to this through this enzyme which is ABHD6, which has been implicated in systemic lupus pathogenesis through different GWAS studies from all over the world. And then we wanted to understand what this enzyme is doing, and we found that this enzyme is highly enriched expression in PDCs, in human PDCs. And then we found that this enzyme is actually regulating PDC function in a great way. So when this enzyme is highly expressed, it cleaves off whatever endocannabinoid comes near the PDCs on the surface. And that prevents the endocannabinoids to affect the PDCs in a negative way. So it was a very interesting discovery made by Wendrila, and we also found that there are systemic lupus endophenotypes where there are ABHD6 positive or high expressor patients where this pathway is extremely important. Now let us come to the more internal regulators. As I already mentioned, the pH in different parts of the endolysosomal compartment has to be intricately regulated by PDCs. Now all phagocytes have their endolysosomal compartment which is acidified, and we know there are VATPases, proton pumps, that will pump in protons inside the endolysosomal compartment that will make it acidified. But in case of PDCs, it requires more control. You will realize that in the last slide of my talk, and because the cargo comes from macropinocytosis and SKICC pathway, the toll-like receptors come from the surface. These vesicles need to fuse together so that the cargo can actually see the TLR receptor in the endolysosomal compartment at the right place. And then the TLR, the proto-TLRs, which are inactive, have to be activated at the right point when it is actually encountering the cargo so that the TLR activation can happen, right? And we indeed found that there are special regulators for endolysosomal pH control in PDCs which are not present in other phagocytes or other immune cells. For example, Purbita Bandyopadhyay, she discovered a polyamine transporter which is present only in PDCs in human circulating immune cells, and they just extrude out whatever polyamines have been taken in by macropinocytosis, and that this extrusion of polyamines from the endolysosomal compartment acidifies the compartment. And also this relates to the polyamine when it is present in cytosol. It mitigates the mitochondrial ROS production, etc. And that also helps PDCs in producing type 1 interferon in a tightly controlled way. Jafar Sharif, another very motivated PhD student, he showed that there are bicarbonate chloride antiporters which are present on PDCs in the endolysosomal compartment. Again, it's not really present in other immune cells. It's previously reported in RBCs where it controls the RBC volume control. And he showed that by extruding bicarbonate and introducing chloride inside the endolysosomal compartment, again it controls the endolysosomal pH in a more like regulated way, so that at different stages of endolysosomal pathway, the pH can be controlled in a more tightly. I'll go a little bit in detail in more recent works which are involving endolysosomal trafficking. I already kind of alluded that endolysosomal trafficking, control of it is extremely important because you are getting cargo through endocytolytic pathways, you are getting TLRs through other vesicles, they have to fuse at some point, and also there are different parts of the endolysosomal pathway. If TLR is activated from that specific step, the functional outcomes in PDCs are very different. And so the classic example of that in PDCs is a differential response to different DNA structure. And it was well known for long, even when I was PhD student myself, that different DNA structures will actually give different functional outcomes, despite the fact that they are activating TLR9 in PDCs. For example, B-type CPG, when you give on PDCs, they are not really efficient, very efficient in producing type 1 interferons. And doesn't work, right? Okay, don't worry. So you can see the B-type CPG will produce more of TNF, IL-6, all these pro-inflammatory cytokines. And interferon production in PDCs are driven by the transcription factor IRF7. B-type CPG fails to activate IRF7 or phosphorylate IRF7 so that it translocates to nucleus. On the other hand, A-type CPGs are great producers of type 1 interferon. They will drive phosphorylation of IRF7. And in a very interesting work back in 2005, Taniguchi's group showed that this difference happens because CPG-B signals TLR9 from the late endosomal compartment. On the other hand, the CPG-A or A-type CPG recognizes TLR9 or is recognized by TLR9 or activates TLR9 from the early endosomal pathway. And that was where the state of the art was. We knew that, okay, the different DNA structure. So here you can actually see the functional outcome is differing because of the cargo size or structure, because we already knew that B-type CPG are usually monomeric, while A-type CPG forms bigger aggregates. And that much we knew. And so a brilliant student in the lab started this program, and it could not be finished by her. Then it was handed over to another student, then another student. It went on for almost 8 to 9 years in our lab just to figure out why this difference in cargo structure is giving different functional outcome in PDCs. And our hypothesis was, well, you can actually see, so the top cartoon shows the model for the monomeric CPG or B-type CPG, which will not produce interferon. And there will be it's a monomeric small molecule that's being taken inside the cell, so the endocytic pit size will be much smaller. And then the A-type CPG, which forms bigger aggregates, naturally the cargo structure will be, much bigger, the structure will be more complex, and that will create a bigger endocytic pit onto the PDC surface. And we thought that maybe something of this sort is being sensed by the cell, and that's how the functional outcome is differing. And as I mentioned, all these 3 students contributed to this, but credit goes to Shrestha Patnaik, who is in the audience, who could actually finish this very intricate question in this story. So our hypothesis was that there is, because of the smaller endocytic pit, the membrane buckling is much less, and there is much less membrane tension being generated on the surface. It's kind of known in the endocytosis field that if the endocytic pit is really small, the membrane buckling will be much less. Even bar domain proteins are activated much less. And we thought that there must be a sensor which is giving this information to the PDC interior so that the functional outcomes can differ. And when the cargo is much bigger, there is higher membrane buckling, there is more membrane tension. And now the question was, what was that sensor on the surface? And we hypothesized that piezo1, which is a mechanosensor. We have come to know about piezo1 really recently, 2010 it was discovered. We started this project in 2014 or something. And so piezo1 is a pure or professional mechanosensor because it doesn't really interact with other proteins to do its function. So it's almost like a Galilean liver, 3 Galilean livers. It's a trimeric protein. And so when there is membrane stretch, these blade types, which are blade parts which are jutting out of the cell membrane comes down, and the central pore opens and bivalent cations get in. And and PDCs, human PDCs, express this piezo1 in a really high level. And it's not only PDCs, also other cells, T cells, B cells, macrophages. Now we know a whole lot of immune cells are expressing piezo1. There are different groups that have come up with new insights on how piezo1 can actually influence immune cell function. Anyway, so our hypothesis was that perhaps piezo1 is doing this, and that means there are bigger endocytic pits being formed in response to this bigger cargo structure. And we did one SEM. You can actually see there are much more membrane ruffling on the CPGA treated PDCs On the other hand, the monomeric CpGB doesn't really produce that much membrane ruffling. By the way, while we were doing this experiment, by that time another group already showed that membrane ruffling on macrophages are actually sensed by piezo-1, and so it actually enthused us. We got into this, and now we wanted to measure whether indeed there is increased membrane tension on these cells. We took help because there are different ways of measuring membrane tension on cells, cell membrane, and you can actually do atomic force microscopy or optical tweezer being introduced to the membrane and looking at how much resistance it's encountering, things like that. But in all these cases, you have to it's kind of interventional methods, right? You are actually poking the cell with something to measure the tension on the plasma membrane. So we are looking for something that will not have this interventional approach, and it will actually not touch the cell, but measure membrane tension from the cell, from the microscopy. And Bidisha Sinha, Professor Bidisha Sinha from IISER Kolkata, who is a physicist who does biology, and she helped us with her microscopy called interference reflection microscopy. We found, so I'm not going into the mechanics of that microscopy, that can actually measure non-invasively membrane tension on the cells. You can actually see on the CpGB, this is the global tension. So this is the distribution of tension all over the cell. But when you are giving CpGA, you can have a higher tension zone that are forming in different parts of the cells. This is more of a global nature of the tension. Now you can actually go deeper and look at the individual endocytic pit by combining TARF imaging and using a cargo which is fluorescently labeled, and then doing this membrane tension measurement. And here you can actually see, for example, here there is a CpGA which is fluorescently tagged oligomer which is entering the cell, and you are also measuring the tension at this region. And this is the endocytic pit, 0 points to the center of the fluorescence, that means the center of the cargo, and then you can actually see there are tension zones forming around the endocytic pit. So that was extremely convincing, and we also saw that when you give CpGA, you start seeing piezo-1 proteins clustering on the membrane, and that means they are doing something. They are being signaled that there is increased membrane tension on the surface. And then we did some of the functional studies. So CpGA should produce a lot of interferon. If you knock down piezo-1 on this human primary plasmacytoid disease, you find much less interferon is being produced. And on the other hand, CpGB should not produce a lot of interferon because it's not able to drive IRF7 translocation. And when you add an agonist, a pharmacologic surrogate for membrane tension, which is an agonist of piezo-1, when you add that, CpGB is also producing interferon, and which is also reflected by the transcription factor nuclear translocation data. Okay, so this recently got published. And so that actually solved almost a 20-year-old enigma why DNA structure can actually drive differential functional outcomes in PDCs in both mouse and human, but we worked with humans. And then I'll just, if I have time, I'll just briefly talk about another endosomal trafficking story that recently was completed by Dr. Ronit Di Rosario. This is unpublished. I'll just talk about it briefly. So Ronit was looking at different proteins or different genes which are highly expressed on PDCs in autoimmune diseases. And in that way, he was looking at all the single-cell RNA data sets from psoriasis, SLE, etc. And he found that PDCs that are flocking into the skin in psoriatic skin, they are expressing a protein called melanoregulin in a really high levels. Now, what is this melanoregulin? So we also checked by GPS data with purified cells, and also we purified PDCs from human buffy coats and looked into melanoregulin expression. Again, it's not really expressed in other immune cells. It's very specific for PDCs. They are expressed in pineal body. They are expressed on melanocytes because that's where they were first reported. So melanoregulin is known to have a dynein-like function in the melanocytes, where it carries the melanophores towards the surface of the melanosomes, melanocytes, and the melanosomes will actually contain all the melanin, and that's how our skin looks brown, etc. And the dynein function shows that it has some carrier function. It can actually bind to vesicles and drive them across microtubules to different parts of the cells. And that was interesting for us. We could not understand what exactly it's doing in PDCs. And so we just did the crudest experiment we do in PDC lab. We just take PDCs, knock down this gene and try to find out what it is, how is it affecting the primary outcome of PDC activation, which is interferon production. It seemed melanoregulin knockdown again inhibited interferon production from PDCs in response to TLR9 activation. More importantly, the knockdown experiments, because these are primary PDCs, you can't really do a lot of other things, but knockdown experiments also gives us ability to have the gene or their transcript in different dosage, right? And that's how you can actually see in this figure, the reduction in interferon is kind of correlated with the reduction in the transcript dosage in different experiments. And more importantly, we found that in melanoregulin knockdown cells, the cargo is less often co-localizing with TLR9. That means the cargo is not meeting its receptor, and which was extremely interesting. Very few people have actually looked into it. I'm not claiming that we have also looked into it. I'll talk about it. It's still on our list, but it's extremely important to know the regulations, how an internalized cargo and the internalized TLRs come together and the vesicles fuse, and now the TLR recognition happens. And so we see that MREG deficiency reduces the ligand receptor engagement. Interestingly, we found they are mostly on the steady state, mostly expressed in the lysosomal compartment, late endosome lysosomal compartment. And if you knock down them, we find that the LC3 recruitment to the lysosomal compartment, fused lysosomal compartment, is reduced. That means it's reducing autophagolysosome formation. If MREG is reduced, right? So that means MREG is driving the canonical path of endolysosomal maturation towards autophagolysosome formation. And perhaps that's how it's also regulating the cargo TLR kind of rendezvous at a specific stage of the endolysosomal trafficking. And interestingly, when you do not have MREG, when you have less transcript of MREG, LC3 starts co-localizing with early endosomal compartment. And that's something very interesting, because you don't expect early endosomal compartments to accumulate LC3, lipidated LC3. And this is known in a specific pathway called liposome formation or LC3 associated phagocytosis happening, and where early endosomal compartments itself, after phagocytosis, there is LC3 recruitment, and instead of going through the lysosomal path, lysosome comes to those already lipidated LC3 kind of studded vesicles, which are actually early endosomes, and then fuses with that and kind of like digests all the material inside that cargo, inside that vesicle. So we thought that, okay, so maybe it's this is happening, the liposome, or it's also called landosomes or LC3 associated endocytosis pathway. And then we had to look at whether this is exactly happening. That was our kind of guess. Now, it is well known in the field of endocytosis and liposomes that Rubicon is a protein, an adapter protein, which is extremely crucial for liposome formation. Rubicon gets recruited into the early endosome, and that starts this cascade of LC3 coming in and bypassing the lysosomal pathway and decorating the early endosomal compartment itself. So we wanted to look at Rubicon, and so now we see that if you make MREG deficient, if you make the PDCs MREG deficient, there is enhanced Rubicon recruitment to the early endosome. So that shows us that the liposome for pathway is being driven when MREG is not there. And can you just reverse it? If you now knock down Rubicon, you start seeing that the early endosomal compartment is not getting lipidated or getting this lipidated LC3 decoration anymore. More importantly, if you use Rubicon siRNA on PDCs, the whatever steady state liposome formation is happening, that stops, and that actually increases interferon production by PDCs in response to TLR9 activation. That means we are seeing a regulator, which is melanoregulin, which was known to be active in melanosomes, carrying melanocytes, carrying melanosomes around inside those cells. Here is controlling the regulation of endolysosomal trafficking and kind of deciding the fate of early endosomes, whether it will take the canonical endolysosomal pathway ending in autophagolysosome formation, or it will take the other path and quickly go to liposomes. And whenever it goes to liposomes, the cargo will be destroyed, and naturally TLR9 engagement will be much less. So what are the questions that remains for us to solve? So we found that PIZO1 inhibits this early endosome to late endosome transition, retains the CPGA in the early endosomes so that it starts signaling from early endosome. And then we find that MREG is actually regulating these two pathways, canonical autophagolysosome formation or the liposome formation pathway. Now the question is whether PIZO1 input is also driving these decision making in terms of which way the early endosome will go, right? Because we already know that, so I didn't show the data, we already know by local actin meshwork formation, early endosomes are retained and they are not maturing. Now the question is whether that is also preventing it from going to liposome and reducing TLR9 activation. Finally, as I already mentioned, it's really enigmatic how the cargo coming through a different vesicle and the toll-like receptor coming through a different vesicle, how they are actually regulated and they come together, and the actual cargo, the ligand TLR engagement happens. And we don't know how exactly it happens. And we are trying to, we are hypothesizing that MREG has something to do with it. So MREG will actually regulate how the cargo will actually meet up with the TLR9 containing vesicles. So I'll just stop here. Thanks for your attention, and thanks for the recognition of whatever small work we have been doing in India. Thanks.
Congratulations, Diploman. And what next? I think coming after Bhatnagar and then this big carrier scientist hour, I think Diploman is going to take the society to a greater height, much greater height. Fantastic work, extremely good and focused. Thank you very much, Diploman. Indian Immunocon Society honors him, and they this honor by recognizing good science, there is a certificate and a check.
Thank you, Professor Dipyaman, and congratulations on a thoroughly deserved recognition. Ladies and gentlemen, we now have the great honor of a keynote from one of the world's foremost authorities on therapeutic antibodies, a scientist whose career has spanned the very century his lecture celebrates. To preside over this session, may I respectfully invite Dr. Savita Kulkarni from BARC, Mumbai. Dr. Faisal Guru from Sher Kashmir Institute of Medical Sciences, and Dr. Nisar Naiku from GMC Srinagar. Please take your seats on the dais. Professor Kaveri Srinivas is a senior research scientist and the Centre National de la Recherche Scientifique, France, one of Europe's most prestigious research institutions. An internationally acclaimed immunologist, his pioneering work has significantly advanced our understanding of antibodies, immunotherapy, autoimmunity, and immune regulation. His research spans therapeutic antibodies, intravenous immunoglobulins, immune tolerance, and translational immunology, contributing to innovative approaches for treating autoimmune and inflammatory diseases. Professor Kaveri has authored 250 scientific publications and holds several patents and translational innovations in the field of immunology and antibody therapeutics. His keynote today is entitled "A Century and Going Strong, the Story of Therapeutic Antibodies." Professor Kaveri, it's our privilege and delight to welcome you to Immunocon 2026.
Presentation. It's okay. Where presentation? Yes. No, I'll find it. I'll find it here. I think so. Yeah. Good evening to all of you. And before I start, I would like to thank the organizers, and in particular Dil Afroz, who is busy on telephone. Yeah, I know. She has a lot of responsibilities. I know. And the organizers for having asked me to come over from Paris to talk to you for some time. And incidentally, I was not really anticipating that I would be talking because I thought I should not steal the slot of a young, exciting immunologist, and why you're asking this elderly person to come over and talk. But then since I'm here, you have no choice. You have to listen to me, okay? One other small disclaimer is I normally aim my talk for the backbenchers because for all my life I have been a backbencher myself. So if the frontbenchers think that it is the same old story, I respectfully accept that comment. This is the group, and thanks to which I'm here, just like Dipyaman said. All the work is done by them, and you come here and then stand in front of you and you talk. And for those people who have been to Paris, you know where we are. Okay, this is Jardin de Luxembourg, right in the heart of Paris. And if you walk at the far end there, you see the Senate building, and behind that is our lab. On the right side is Panthéon, where great lives, including Voltaire, Guy de Maupassant, not Pasteur, not Curie, and those many other scientists. They're there. And these are my scientists, and they're quite an international group, and they're having a lot of fun. And some of them are here actually. Vani is here. Vani has come back to India, but today she's not here. Jagdish, And I see Vandana is here, yes. And the top, the panel there is all the people that came to my lab, students from one institute in India. There's no bias. I'm from Bangalore, by the way. So they are from Indian Institute of Science, but no bias, no bias. Sometimes these people are also in the lab. And when they're in the lab, they're quite good, okay? So just like Deepaman said, we also have to collaborate. You know, we are asking a lot of basic questions and clinically oriented questions. I'm only a veterinarian. Nobody's perfect, right? So I work a lot with clinicians, and these are the clinicians that help me all over France on the left side here, and quite a few collaborators in India, elsewhere, and I'm sure some of us can recognize the group of Nagaraja, Balaji, D.N. Rao in IASC, Kanjaksha Ghosh, Deepika Mohanty, Veer Singh Negi, who is now in Jipmar, back in Jipmar. Jipmar, Pondicherry, yeah. Anyway, I collaborate with a lot of people and I thank them. So in a very simple-minded view, when we confront a pathogen which comes with either, damp, danger-associated molecular pattern or pathogen-associated molecular patterns, they are recognized by the innate immune compartment and through the pathogen. Speak up loudly? Okay. They are recognized by the pathogen-recognizing receptors, also called Toll-like receptors, and in principle, most of the low level of infections are taken care of by the innate immune compartment that you have studied in your high school. However, if the innate immune compartment cannot cope up with the pathogen, either because the virulence is very high or the pathogen load is very high, then you kick in the adaptive immune compartment. And in the adaptive immune compartment, just like Deepaman said, antigen-presenting cells play a very crucial role. They pick up the pathogens, they chop into pieces, they cut these peptides in, they show them, they present them through the class 1 or class 2 molecules, depending on the nature of the antigen, and they show them to the T cell receptors, and the T cells does get activated and start proliferating. And some of these T cells, they provide signals through the cytokines to B cells. B cells that are dormant now become plasma cells and start secreting antibodies. And antibodies, unlike the innate immune compartment, are more specific, as you all know. And through their variable region, they can recognize the pathogen. And imagine the pathogen has already reached the target organ, let's say the lungs here. The association of the antigen with the antibody initiates a whole cascade of events. The FC portion of the immunoglobulin, you all know, antibodies are Y-shaped molecule. You have the variable region that binds the antigen. The antigen is in the lungs now, and the constant portion, the FC portion, engages with the FC receptors that are present on the macrophages that you see in red there. And engagement of FC receptors on the macrophages initiates a cascade of events that are inflammatory. And this inflammatory, as you all know, is a healthy inflammation. We need that because that tries to get rid of the pathogens. And if that's not sufficient, the neutrophils come into picture, the ROS molecules are secreted, some other obnoxious molecules are secreted like nitric oxide. The idea is to get rid of the pathogen. And in principle, it works mostly. But if it doesn't, then you also kick in the complement cascade. And the complement, as you may know, at the end of the complement activation, you have membrane attack complex that sits on the pathogen, that drills a hole, the pathogen is killed, lysed and killed. That way, we try to eliminate the pathogen efficiently. And if that also is not sufficient, then the T cells get activated in a very well orchestrated manner. The T cells now differentiate either into Th1 or Th2. Th2s regulate the Th1 if there is excess of Th1. And we have also understood the role of Th17 cells, and they are controlled by the regulatory T cells if the Th17 cells become uncontrollable. All this leads to the elimination of the pathogen, and that's a good thing. And as we are sitting here, that's happening all the time, okay? And in addition, we have a bonus. The bonus is some of these T cells become memory cells. They're sitting there. And some of the B cells also. So if you are exposed to the pathogen in the due course of time, these memory cells get activated very quickly and they shortcut and they provide the help very soon. And this, as you all know, is the principle behind the vaccines. Okay, you want to have as much as potent memory cells as possible. That's the principle of vaccination. Now, if everything goes well, it's fine. I mean, all these components are in place. But unfortunately, several conditions have been identified wherein some of these components can be missing. Yesterday, I'm sure you heard Madkaikar, Manisha, and she would be telling how many exactly are now identified, how many conditions, how many genetic defects have been identified that can be responsible for primary immunodeficiency. In other words, if any of these components that I told you in my this poster, if any of these components is missing, then you are immunodeficient, which means you will not be able to fight the pathogen as you should, and you will be susceptible for infection and sometimes fatal. And this genetic condition is called primary immunodeficiency. My respects to Alain Fischer, one of those pioneers who has been trying to cure these genetic disorders through gene therapy. And my interest in this is only a simple molecule called antibodies. And this I have been studying for some time. But the story goes back to long back. You know, this is my journey starts almost 120 years ago, although I was not there. Emil Adolf von Behring, along with Kita Sato, identified some molecules in the serum or plasma of the animals that were injected either with a toxin or inactivated pathogen. And when he looked at the serum or the plasma, he could use that serum or plasma to treat another animal that was challenged with infectious agent, either Clostridium. In first case was tetanus, and soon they discovered that if you could do that with diphtheria, you could actually cure diphtheria, meaning you raise antibodies and you can use these ready-made antibodies to cure diphtheria. And I'll tell you, by the way, we are talking about 1890s, and there was no other treatment. If you had diphtheria, the child would die. There was no other cure. And for the first time in the history, first passive immunotherapy was practiced, and first ever Nobel Prize was won, and soon hundreds of children were saved, thousands later on. And in Paris, across this side of the river, Émile Roux, a disciple of Louis Pasteur, himself treated in Necker, Hôpital Necker. Some of you may know this hospital. In that hospital, he treated more than 300 young children and cured them of diphtheria. This now became a central topic, major interest because we knew what is this magic bullet that is present in this ampule. So people started looking at the functional aspects of these molecules. They started identifying what exactly is this molecule, structure and binding properties, their affinity, avidity, those aspects. More interestingly, by then, Louis Pasteur had happened. We knew how to identify a pathogen. We knew how now to make antibodies or serum against these pathogens. And people started making antibodies against many pathogens because, mind you, we didn't have antibiotics at that time. No penicillin. These people, Robert Debré, Charles Nicole, Ernest Conseil, most of them French, all of them French, they identified all these pathogens and then prepared the serum. And now we had in our hands antibodies that were ready to use for passive immunotherapy. Many Nobel Prizes were won, many other discoveries were made. A complement was discovered, anaphylaxis was discovered, antibodies, autoantibodies to blood group antigens were many decades passed. I'm going fast now, fast forward. But the bad news came. The bad news came in the form of the World War II. During the World War II, the soldiers were dying not just on the battlefront, they were also dying in the hospitals because of infections they would have carried or because of the hemodynamic issues due to blood loss. And it is at that time that the President of the United States of America commissioned a certain Edwin Kahn, who in Boston had already mastered the technology of plasma fractionation in a very high way, industrial way, very sophisticated manner. And Edwin Kahn could identify all these different components, and one of the major components, as you might have, whenever you order albumin, you still order it as cons fraction 5 actually. Even today it's called cons fraction 5. Just check it out whenever you order albumin. But there is antibody fraction. And those antibodies, they helped to treat thousands and thousands of soldiers were saved thanks to these antibodies. And the plasma, the albumin saved because of hemodynamics issues. So plasma industry started flourishing. It is just about that time, having returned from the World War II, Colonel Bruton, Ogden Bruton, he had come back from Europe and he was practicing in the East Coast. In one fine day in early 50s, he gets this young boy who is not responding to any treatment that we knew. He was having repeated infections, repeated pulmonary infections, sinusitis, and other respiratory issues. Bruton didn't understand what's going on. Why is this boy? By now, we had antibiotics. By now, the penicillin was discovered. Alexander Fleming had happened. Even the antibiotics could not help this boy. And then Bruton thought, maybe there's something innate problem with this boy. Let me check. And he looked at the normal profile on the very top of a normal, healthy human being. And then in the mid panel, this particular boy did not have the gamma globulin fraction at all. And in the bottom, when he was injected with he asked Malcon, send some antibodies, I'll inject this boy, let me see what happens. At that time, it was a thick pellet, and actually it's my emulsion. And he injected it to this boy. In 5 days, the boy was kicking around, jumping around. He had just recovered from some of the infections. And this was a dramatic discovery. Two things happened. One, we knew that there are conditions in children. They can be born with these conditions called primary immunodeficiencies. We can identify them. And two, we can treat them. They were dying. Earlier they were dying because there were repeated infections. People were not identifying what exactly it is. They were trying all antibiotics, but nothing was happening. But that discovery changed everything. And now we could treat these immunoglobulins. Except this was a paste, as I said, into a thick solution. And Charles Genevay, I'm sure you have heard this name, Charlie Genevay, he treated this immunoglobulin paste with pepsin in acidic conditions, and he treated himself through intravenous route, and he showed that you can tolerate these immunoglobulins, and actually it worked. Meaning he could be treated, one could treat with intravenous route. Earlier it was only intramuscular, it was painful, there was gangrene, there was abscess, and it was very painful. So discovery of immunoglobulins that can be injected through the IV route was a major discovery. My respects to his son. What genes, huh? You have father and son. You know, some of you know Charlie Genevay Jr. I'm sure you all have read the books of Charlie Genevay, okay? My respects to him. Now, IVIG is on the scene. Just for you to know what IVIG, let me define because I'll be using for the next 3 hours. I'm kidding. Because there is dinner in between. Therapeutic IVIG is intravenous immunoglobulin. Intravenous because not it comes from the veins, because it goes to the veins. Intravenous immunoglobulin. And because it is purified from several thousand healthy people like you and me, it contains antibodies, like we all contain antibodies to external pathogens, external antigens. We also have antibodies against self-antigens. We'll talk about that on some other day, the natural autoantibodies. But there are also antibodies against antibodies called anti-idiotypic antibodies. I may mention this a little later. So we have this preparation, and we started identifying. First condition was identified, which was A-gamma globulinemia. Soon, all these other conditions of immune deficiencies were discovered. Now, we know more than 150 or maybe close to 200 conditions that can be identified as primary immunodeficiencies. Not all can be treated with IVIG, but most of them can be treated with IVIG. But we have other treatments also. Some years fast forward once again, 80s, strange 80s. On one cold October, drizzly, depressing, cold, cloudy morning, I land in Paris. Nobody noticed that. But luckily, I met this person. And I don't think you're looking at the right person. In fact, I have been told by many of my senior friends who share the same hairstyle as me, meaning more style than hair, who are sitting in the front benches, they come to listen to my talk, or they come to my talk only for one slide. It is this one. Anyways, that's a confession between us. I met Michel Kazachkin, and Michel was a very interesting person. By then, just then, HIV was discovered, and he used to go all around, and we were treating, he was also interested in secondary immune deficiencies, and something equally interesting happened at that time, and that was Paul Imbach. Paul Imbach happened. Paul was a pediatrician too, and he was practicing in Inselspital in Bern, and one day he gets this patient, a young boy who had autoimmune thrombocytopenia. As you may know, in thrombocytopenia, autoimmune, you have autoantibodies directed against glycoproteins of platelets. When the autoantibodies opsonize the platelets are phagocytosed by the macrophages, and therefore the platelet numbers go down dramatically. Curiously, fortunately or unfortunately, the boy also had hypogammaglobulinemia. And Paul did not know what to do. He has an autoimmune condition, but he also has a hypogammaglobulinemia. What he knew that we can treat hypogammaglobulinemia with IVIG, because by then IVIG was well mastered. They were treating that IVIG with IVIG, many hypogammaglobulinemia condition. So he just gave 400 milligrams of IVIG, and what happened is a dramatic thing. He was treating hypogammaglobulinemia, but the results that he obtained was in 4 or 5 days, the platelet numbers were back to normal. He didn't understand, is this fluke, something, serendipitous? It may not happen in all the patients, but he went on treating several other kids, several other autoimmune patients, immune thrombocytopenia. All of them had this beautiful result. And it is at that time in our lab, we were treating, we had, in adjacent group, autoimmune hemophilia. Because platelet disease is associated with coagulation defects, we said, why don't we treat these patients with IVIG? These are autoimmune patients. I'll tell you, autoimmune hemophilia. These are women in their 3rd trimester of pregnancy that develop autoantibodies. This is not autoantibodies. These are not boys X-linked. This is not the congenital hemophilia. This is autoimmune hemophilia. And when we treated those ladies with IVIG, the same treatment, same dose, in 4 days, their autoantibodies levels came down, and they went on to deliver babies normally. And that means we've opened the floodgates. And in rheumatoid diseases, they started using, and in some of the ophthalmologic conditions, for example, in autoimmune Graves' ophthalmology or autoimmune uveitis, IVIG was used. But I shouldn't say worst thing. The crazy thing was with the immunologists, neuroimmunologists. They started using, with perfectly done, well done, proven effective studies in clinical studies, clinical trials, Guillain-Barré syndrome, CIDP, and many conditions, proven effective. And in probably effective, myasthenia, polymyositis. But curiously, neurologists, I shouldn't say that actually, neurologists went crazy. It's not nice to say, right? But they started using in all these different conditions, in Alzheimer's, in Parkinson's, in autism, schizophrenia. In fact, one of these meetings we were asking off-label use of IVIG. Irvin Gelfand, he said, we will use IVIG in chronic fatigue syndrome. And then I asked, Irvin, why do you want to use in chronic fatigue syndrome? And the answer was, why not? With that accent, of course. But then why not is not the right thing? And because of this, use of IVIG rather indiscriminately, the present use of IVIG is so high that in 2030 or so, we will be reaching an industry of 25 billion euros. That's a lot. But the problem was, if there is a scarcity, do we have an alternative? What do we have? Right now, nothing, for these different conditions. And the mechanisms action were not well known. And this is when we took up the action of understanding the mechanisms of action of IVIG. In fact, IVIG is such a simple molecule, right? You have immunoglobulin. It's a variable region, constant portion, and the constant portion sits on the FC receptors, as I told you earlier. And the FC receptors are present on so many different cell types. And in most of these FC receptors, gamma R1, R2A, R3A, R2, those are all in several different important functions, mostly positive, mostly pro-inflammatory. But there's one receptor, Fc gamma R2b, that carries a ITIM inhibitory motif. And that, if you engage that, you actually inhibit the auto-inflammatory or inflammatory process, just for you to know. So we went on, started looking at different mechanisms of action of IVIG. One thing that we discovered was, okay, there are idiotypic antibodies, and I don't know whether some of you, the seniors, would know, but juniors don't even study anti-idiotypic antibodies. These are antibodies that are directed against other antibodies, okay? And they can neutralize the first antibody. This was a Nobel winning observation. We understood that there are some anti-idiotypes in your immunoglobulin preparation, but we knew that inflammation is the hallmark of autoimmune diseases. So we started looking at how IVIG can modulate the complement activation, cytokine network, the TH and TH2 balance, beautiful results. And then came Jeff Ravitch. Jeff Ravitch said, forget it. You close your shop, Srini. I'll tell you how it works. Forget all the theory that you made for 20 years. IVIG works because there is only FC portion that is important there. And not even all FC. Only 8% of your FC that are actually having asparagine in 297 position. And that's all. You extract that. You make either monomers, trimers, and it is sufficient enough to treat all these conditions. That's his observation. And he published some silly papers in silly journals. You can see that. And while he was doing those silly papers, Jagdish came to our lab. And Jagdish, very innovative. And Jagdish is now in IIT Palakkad, by the way. He is head of the biological. Jagdish thinks originally, thinks differently. And we knew by then the dendritic cells play a very important role in the regulation of T cell functions, T cell polarization, B cell functions, etc. So he started looking at that and demonstrated that IVIG can actually block the differentiation, maturation, and function of dendritic cells. And this was a little bit shocking for us, but very interesting, okay? It's very interesting because if you induce the tolerogenicity to dendritic cells, you also induce regulatory T cells. And we know how important are regulatory T cells. I don't have to tell you how important are Tregs. It is at that time Shiva Shankarvati, Lord Shiva, he came down to earth, sometimes he does. And Shiva in our lab, he comes from Bidar in Karnataka, by the way. He showed, not only in vitro, but in vivo models, also in human patients, in Guillain-Barré syndrome, that when you inject IVIG, you downregulate Th17 cells and upregulate the regulatory T cells. Showing that was one thing, but exploring the mechanisms was more fun. We took some years, but we explored one after the other, lots of interesting underlying mechanisms. It's dinner time, I don't want to go into the details. So we skip the details, but I'm ready to answer the questions. Right now, what we, in fact, one second on that, because I didn't go into the details, but IVIG has a capacity to suppress the Th17 cells. We know what are the underlying mechanisms, expand the regulatory T cells, and we know what happens there, underlying mechanisms, and in turn, how Th1, Th2 balance can be maintained. This all happens through the entire molecule of IVIG. And the so-called monomers, trimers, or hexamers of Jeff Ravitch, they're good. I mean, as I said, fantastic papers, but we still have not seen them in clinical practice, at least not yet. So all is not all that beautiful in IVIG, because all the patients that we treat with IVIG do not respond the same manner. About 70% of the patients, for example, in CIDP, chronic inflammatory demyelinating polyneuropathy, they respond. But 25 to 30% of the patients do not. And it would be good to know why these 30% of the patients or 25% of the patients do not reply or do not respond. And to understand the markers, if there are any, it would be very important. And so, right now, we are trying to use machine learning or artificial intelligence. I'm a fashion victim, L'Oreal, you know. So, we are looking at transcriptomes and see whether we can identify those individuals who have a signature that respond and those who do not respond. And if we can identify them beforehand, you can save a lot of burden both on their immune system and on their financial situation. I must have convinced to you that now natural antibodies are important for immune deficiencies that you all knew.
Sir,
You didn't need me
sir,
for that.
2 minutes.
I'm seeing 40 seconds. over there.
Okay.
39 seconds. Now, you took away 2 seconds from me. But I also hope I have convinced to you that these normal immunoglobulins that are circulating in you right now, they have potent immunoregulatory functions, and we still don't know everything about these very normal immunoglobulins that are there in you and me. And one last slide. The way I look at the immune system is this. Perfect day in Srinagar. Dal Lake, peaceful, serene, beautiful sunset, beautiful sunrise. Everything is fine. All the mountains around here, the Pari Mahal, etc. Beautiful Srinagar. This is our immune system on a beautiful day, on a perfect day. But when one clone or one molecule or one cytokine starts misbehaving, one autoantibody, you think it is one molecule, one cell, one... No, it is actually much more than that. You disturb the entire architecture of the immune system, and we have Fourier transformation studies, mathematical computations to show such a thing happens. And if such a thing happens, meaning you disturb the entire architecture of the immune system, we believe IVIG, by bringing all the physiological elements, readjusts or brings back the immune homeostasis to a normalcy. And this is how my wishful thinking, this is how it should work. It should be working. But I'm yet to be, real demonstration. Thank you very much, and thank my colleagues.
Sir, for your interesting talk. Like it was really, I'm sure that all of the audience enjoyed the talk the way it was, and especially students, from the way it started with giving everything from like how exactly everything started. So now it is open for any questions.
Yeah.
Any audience? Yes.
Please.
2 questions.
You can shout.
So just a short question. What would be the use of IVIG for type 2 diabetes? Have anybody tried that one, and have they shown reduction in insulin resistance and things like that?
That's a very good question. Yes, there are papers from Swedish groups for the use of IVIG in type 2 diabetes, but unfortunately they could not reproduce. There were beautiful animal models also, the NOD models, and there are many other models of diabetes. IVIG has been tried, and hypothetically it should work. With all the beautiful stories that I told you, why it's not working, we do not know. And this is exactly why I think the transcriptomic studies or the big data studies are important to identify, are there some signatures in some diseases that we can spot and say, all right, these patients, these conditions, don't use IVIG, there's no need, because IVIG is extremely expensive and it's a big burden because you are giving almost 1 to 2 grams per kilogram of IVIG. Answer to your question, it has been tried, but not much with success. Yes, please. No?
If there are no more questions, I thank Professor Kaveri for this extraordinary talk, which it was a journey through a century of therapeutic antibody science. We now move on to the felicitation. May I request our worthy chair, Dr. Savita Kulkarni ma'am, to kindly felicitate our speaker of the evening, Professor Srinivas Kaveri. May I now request Professor Srinivas Kaveri to felicitate the worthy chair, Dr. Savita Kulkarni. May I now request our worthy chair, Dr. Savita Kulkarni, to felicitate worthy co-chair, Dr. Nisar Naiku. Thank you so much. We now move to a plenary lecture at the cutting edge of HIV cure research. a field where immunology, virology, and clinical science converge in the pursuit of one of medicine's most important goals. To preside over this session, may I respectfully invite Professor Satya Das from the All India Institute of Medical Sciences, New Delhi, and Dr. Mahrukh Hamid Zargar from the Sher-e Kashmir Institute of Medical Sciences, Srinagar, to kindly take their chairs. Professor Vijay Kumar Velu is Associate Professor at Emory University and a globally recognized expert in HIV immunology, vaccine development, and immunotherapy. He is renowned for his pioneering work on PD and PD-1 blockade technology to enhance antiviral immune responses and has made significant contributions to HIV cure research. He has more than 150 peer-reviewed publications and holds multiple patents. Professor Velu is a recipient of the Emory School of Medicine Researcher Appreciation Award 2025 and currently serves as Vice President of the Infectious Disease Society of India. He's going to talk on cytokine therapy for HIV-SIV cure strategy. Professor Velu, we are delighted to welcome you. The podium is yours.
Thank you. Thank you, Azir. Thank you, the organizers. Thank you, Amit Avasti, and all my friends here. Thanks for inviting to give a talk on Immunocon. This is my first conference. It's been a while that I want to come here, but this time, in fact, I made this. This is my short bio. In fact, it's been told before. So I'm going to talk on cytokine therapy for HIV and SIV cure. I have 22 years of experience in working on HIV vaccine development therapy, and I'm an associate professor in the Emory Vaccine Center, and my gurus are Rafi Ahmed and Ram Amara. I did my postdoc with Ram Amara. Again, thanks for the Immunocon for inviting me. So this is the outline of my talk. This is, thanks. So first I'm going to talk about the problem. That the problem is, one of the problem in HIV is that TFH cells, and I'm going to tell about the characterization of the TFH cell and the impact of these cells. These are the reservoirs. And second, I'm going to talk about the solution. And again, the cytotoxic T cells are important, particularly during the viral infection. I'm going to talk about two cells, cytotoxic cells. One is CD8 T cells and NK cells. And then I'm going to talk about a novel strategy for HIV cure, where we found something interesting, that cytokine IL-15, IL-15 receptor for sustained viral control. Again, this is the time duration we work for it. In fact, we started working from 2005, but then we found something, TFH, or the solution and the strategy during the time course, what I told you. So more than 40 years, there is no years of HIV science. There is rapid progression, but there is no vaccine or no cure so far. Although, as you can see, in a lot of interventions, but still we don't have a good vaccine or a cure strategy.
Dr. Vijay, can you speak closer to the mic, please?
Is it okay now? Yeah. Wow. Okay. So there is no vaccine strategy or a cure strategy. So this is estimated people living with HIV. 42 million living, and it's almost 30 million died. About 60 new infections per minute. And again, by the end of my talk, you might know that how many people would have been infected with HIV in the world. We need an effective vaccine or a curative strategy for HIV. Again, we have standard art, and then HART is doing well. And the problem, they reduce virus, they reduce the risk of transmission, they actually prevent HIV-associated complications and with all the other comorbidities. But still, it generates art-associated complications when taken for a long time. Another thing, the heart does not, as you can see in the figure, heart does not eliminate latent reservoirs. So as you can see here, if somebody is put on ART, if you stop ART, the virus comes back to the same level. That's the problem for HIV. Again, ART interruption brings the virus back as soon as possible. Very soon, within a month or so in humans. And another problem is the size of the infectious HIV reservoirs. It is stable over time. This is a study from Johns Hopkins where they followed people who are on ART for many years. In this case, it's almost 7 years. As you can see here, there is no reduction of viral reservoirs, although they have been treated for 7 years with very high intensified antiretroviral therapy. And again, the half-life of these cells are like 44 months. Again, 1 in 1 million cells of CD4s are the reservoirs. And they are very slow decay. You have 4 billion CD4 T cells in the body. Another thing is that they reside in different anatomical sites. For example, in this case, is brain, lymph node, gut, and bone marrow. So they are 1 in 1 million, but they also reside in different tissues where you cannot push CD8 T cells to kill this thing. CD4 T cells are considered more than 90% of HIV as well. But one subset that we found of CD4 T cells are enriched during chronic infection. Although we know that the CD4 T cells are depleted during HIV infection, one subset actually persists or enriched during HIV infection is the TFH cells. As you can see, the CD4 T cells in the pink dot line, they decline after the infection, but the green one, the green dotted line, that's the TFH cells, they actually enrich in one particular place, that is the germinal center TFH. Again, you can see the duration of time. And when you talk about CD4 T cells are heterogeneous, antigens, cytokines, location, master regulator, transcription factors, and they are very plastic. As you can see in this, we have TH1, TH2, 17, 9, 22, Tregs, as well as TFH. Again, TFH is unique. They are B cell helpers. They help B cells, and they make antibodies. Any infection comes in, they get rid of them by making antibodies for that particular antigen. So almost all pathogen-induced antibody and TFH are unique B cell helper. So what's the problem here in HIV? So you can see in the left-hand side, where during normal infection, when there is an infection, there is an antigen impulse, there is, the antigen has been taken to the lymph node, where the TFH talk to the B cells, they make antibodies, they clear the infection. But during chronic infection, the same thing happens. One thing that HIV exploits is that they seek refuge in B cell follicles. The B cells follicles is known for two particular subset of cells, that is B cells, as germinal center B cells and germinal center TFH cells. But there is no CD8 T cells, particularly cytotoxic cells or NK cells. Okay, so there is no access for these killers to go and kill the infected cell. Then actually they stay in germinal center. That's the reason you cannot get a cure for HIV. Okay, and then what we did, I've been working in the primate center for a long time, and we use this SIV model, where this particular SIV model mimic chronic HIV infection. So they have a similar pathology, similar CD4 depletion, similar viral dynamics, and again similar level of seeding of reservoir. So we use this model to really look at, to see what is happening for viral control or viral reduction or viral persistence. Okay, so what we found back in 2014, I guess we published this in GI, where after chronic infection, there is one subset of cells in the lymph node enriched. So you can see at week 0, week 3, and week 14, the TFH, so-called CXCR5 PD1 positive cells actually increased. And again, if you look at between the plasma viremia, between the healthy controllers as progressors as a lot of antigen, all these progressors all have higher TFH. Okay, and then this TFH correlates with the plasma viremia. When you have TFH, high number of TFH in the lymph node, you have high levels of virus in the plasma. Okay, again, when we were talking about that, there are people have published in JXMAD human data. They showed a similar like what we showed, and again, we also showed, particularly my work with Ramalab actually showed.
Can you become a little louder?
Yeah, okay. So we showed that the TFH1 cells, although there is TFH, there is a heterogeneity within TFH, they express CXCR3. We found that, and again, those cells express high levels of viral DNA. So to conclude, what I showed here is there is an accumulation of CD4 T cells in the zone where is color yellow, that is germinal center, but wherever you see the depletion, you see only in the non-germinal center area. Okay, when HIV get infected, that the CD4 goes and resides in the germinal center follicles. They stay there because they cannot be killed, because there is no access, poor access by cytotoxic cells. This is the conclusion of part 1. So the role of cytotoxic cells, how we'll end up in finding cytotoxic cells, again, we all know that initially, many studies that the germinal center lacks cytotoxic cells, particularly CD8 T cells. Then we wanted to see what is the role of it. How do we found it? So this is a study by Rama's lab. At that point, Rama vaccinated animals, challenged them, as a control animals as well as the vaccinated animals. And in the bottom panel, as you can see here, there are a lot of animals got infected right after the vaccine. But if you look at this, there are some animals, they control the virus. There are some animals, they did not control the virus. We went on to look at to see whether what is the difference between the one that's controlled and not controlled. When you looked at the lymph node particularly, the quality of lymph node CD8 T cells are different. They do express CXCR5, as you can see in the figure A. And during chronic infection, you have an expansion of follicular CD8 T cells, and then it is highly expanded after chronic infection. And then when you look at the quality in the lymph node, you see higher expression of CXCR5 positive CD8 T cells, both in the blood as well as in the lymph node. When we look at the correlation with the viral load, wherever you have high levels of CXCR5 positive CD8 T cells, you do see lower level of virus. But that is not happening in CXCR5 negative CD8 T cells. And just to confirm, we stained for CXCR5 positive CD8 T cells in the lymph node. As you can see here, the tetramer positive cells are enriched in the follicles where you see a very low viral load. But the animals that had very high viral load do not have CD8 T cells in the follicles. And as you can see here, again, there is a rapid reduction of these cells when you have CXCR5 positive CD8 T cells, both in the viral RNA as well as msRNA, suggesting that it also have an impact on viral reservoirs. And again, we published this in PNAS along with Rafi when we published paper in Nature. And then since we have two subset of cytotoxic cells, CD8 T cells and NK cells, we went ahead and looked at to see whether is there any role of NK cells in the B cell follicles. Again, we took this type of animals that have been infected with CHIV. We went on to look at to see whether the low viral RNA level animals express high levels of CXCR5 positive NK cells. When we look at it, as you can see here, there is a significant high levels of CXCR5 positive NK cells during chronic infection, and there is no expansion of CXCR5 negative NK cells, although there is a total NK cells expanded ones on CXCR5 positive cells. But when we looked at the same time with associated with the virus, the CXCR5 positive NK cells actually negatively associated with the virus, meaning higher levels of CXCR5 positive NK cells, lower levels of virus. And again, this also associated negatively with GCTFH, meaning the cell that carry reservoir is actually negatively associated with the CXCR5 positive NK cells. We also showed that they are in the follicles by staining them for NK cell marker in monkeys. As you can see here, there's a highly expansion of CXCR5 NK cells. At that point, this is a paper that I was a corresponding author. At that point, what we did is we sorted NK cells and then we did an RNA-seq. What we found was the IL-12 and IL-15 signaling was highly upregulated on these NK cells. And then when we treated them, we went on to do an experiment to treat them with the cytokine, particularly either IL-12 or IL-15 or in combination. As you can see here, the granzyme B positive NK cells are expanded when you give cytokines like IL-12 and IL-15 in combo. You see this proliferation of cytotoxic NK cells, also expanded in vitro. This is the lymph node cells, and then we did this in vitro. And what we did is after this, again, we published this where we said that whenever you have NK cells that express CXCR5, they actually tend to have, the animals tend to have low viral load. Again, at that point, number of studies suggested that IL-15 actually drive CD8 T cells into B cell follicles by many people, including our study. One other study is that this Nature Medicine study where they also said that the NK cells are enriched in animals which have low viral load, particularly in the non-pathogenic SIV model. So that actually kind of intrigued us to see what is happening in our model. So what we did is we quickly wrote an R01, myself and Rama, and we wanted to do the study in vivo, what we did in vitro, to see whether we mimic the same result. In this case, what we did is we took 4 groups of animal. In first case, we gave SIV and antiretroviral therapy. That's a control group. In second group, we gave SIV, and then we gave 2 phases of IL-12 treatment. And in third group, we gave IL-15 and IL-15 receptor treatment, 2 phases. And in fourth group, we gave both IL-12 and IL-15. And we gave them 10 microgram for IL-12, 20 for IL-15, and 100 microgram for IL-15 receptor alpha. And this is subcutaneous infection. And the reason why we wanted to give 2 phases, in the first phase, we started when it was not on hot. At week 6, they were not on hot because whatever the therapeutic intervention you do during chronic infection, you need to have antigen so that the CD8 T cells can expand. If we put down antigen, and if we give any therapeutic intervention, the CD8 T cells don't work. So Rafi suggested this, then we went there. We gave the cytokines in the first phase so that whatever expanded in the first phase can be regained during the second phase. That's the plan we had. So what I'm showing here is, as we expected, look at the green one as well as the red one where you have IL-15 or in combo. Those animals, this is the mean value of, each group has at least 5 animals. This is the mean value of CXCR5 positive CD8 T cells. You can see the dynamics of this, temporal dynamics of the follicular CD8 T cells. In the left-hand side, you see blood. In the left-hand side, you see you are seeing the blood, and in the right-hand side, you see the lymph node data. You see higher levels of CXCR5 positive CD8 T cells expanded in blue and the red group, not in the black one or the blue one. And we also looked at to see whether there is an expansion of NK cells. As you can see, the NK cell expanded. And particularly interestingly, the expanded NK cells are CD16 positive NK cells. CD16 positive cells are known to be ADCC functioning cells. As you can see here, they are expanded during the ATI time point, both in the blood as well as in the lymph node. And here I'm showing the dynamics of the viral dynamics. If you look at the black one or the blue one, during the first phase of the cytokine therapy, there's no much happen. The dark line indicates the geomean of the virus. And if you particularly look at the IL-15, right after the cytokine treatment, the viral actually went down. When you look at the IL-12 or IL-15, there is not much different. And during the phase 2, in the bottom panel, usually when we give cytokines, there'll always be a blip of virus that other people have published. We didn't see the blip of virus because although we are giving cytokine, I don't think we gave a very high dose of cytokine. We have given the optimal dose of cytokine, so there is no much blip on the virus. So here again, the viral dynamics post-art interruption, you see the black one. Look at the virus dynamics. The dark shaded line is a geomean of the virus. See the control and see the blue one, not much difference. And if you look at the IL-15, the very first thing you can notice is right after you stop the antiretroviral therapy, the virus came back, but it did not come back compared to the one that happened in the black or the blue or in the red, meaning there is a blunt in peak viremia post antiretroviral therapy interruption. And again, if you look at the set point, again, you see the set point viral load after 20 weeks of follow-up, the virus has not gone very high level compared to either the IL-12 or in the combo. One of the reasons the combo didn't work very well is that one particular animal, as you can see, they have very high viral load. Again, this is the data, I'm just showing it. Because of that, probably we might have missed it. On the other hand, we also saw that the most of the effect is actually on the IL-15, although it was in a combination group. So what we figured out, this is in fact, in my career of this 22 years in training of an SIV and HIV model, I never seen a data in blunt in peak viremia, and then the blunt in peak viremia post antiretroviral therapy. One other thing I wanted to stress at this point is that compared to the black and the green, you see almost 2.5 log reduction of the virus. particularly in humans. If you reduce 1 log of virus through any therapeutic intervention, meaning those HIV-infected individuals will live 5.5 years longer. In this case, I'm showing 2.5 log reduction, meaning if it is happening in humans, they at least live more than 10 years. If you look at India and curve, there is a significant p-value in IL-15 treated animal compared to the control. At the same time, the fold reduction of the virus is very high in IL-15 compared to the control. Then the question is, what is the mechanism? We wanted to see what would be the mechanism for this. As we thought, as per our hypothesis, we looked at to see whether this CD8 T cells are in the follicles. Again, in the bottom, in panel A, B, and C, you're looking at the tetramer positive CD8 T cells. They are granzyme B positive as well. They are also cytolytic. Okay, and in the middle panel, you are looking at the area where it's supposed to go. That is a germinal B cell follicles. As you can see, the green one, the CD8 T cells are highly enriched in the B cell follicles. Again, they are highly enriched for cytotoxic cells, both in the B cells follicles. Again, we also saw that these CD8 T cells is also expanded in the T cell zone. If you look at the extreme right panel, you see that the green the bars, you can see that they are very high in the B cell follicles. What we did to see whether is there any association of these cells, whatever it elicited during this therapy for the viral control, what we found is particularly the CXCR5 positive CD8 T cells in the blood. At the same time, these CD8 T cells are all also enriched for co-stimulation potential. They also express CD28. And we also see the blood CD16 positive NK cells correlated negatively, okay, with the virus, suggesting that when this therapy, particularly the IL-15 therapy, actually enriched CXCR5 positive CD8 T cells, one, and then they increase the quality of CD8 T cells, that is more towards central memory phenotypes 2, and it also expanded NK cells, particularly the cytotoxic NK cells that is responsible for ADCC. And this is same true for lymph node. As you can see in the bottom panel, for CXCR5 positive CD8s or CD28 positive cells or the central memory positive CD8 T cells are associated negatively with the virus, suggesting that enhancement of these CD8 T cells and trafficking of these CD8 T cells to the B cell follicles kind of put down the virus in these animals. And to conclude again, with this therapy, IL-15, particularly IL-15 therapy, elicited functional CD8 T cells and NK cells. They somehow trafficked to the lymph node. They actually cleared the infection, particularly looks like they killed the TFH, and then thereby they reduced the antigen in the lymph node, and then they reflected in the post-ATI time point. Again, the interesting thing that this could actually benefited is that they reduce the peak viremia as well as they reduce the set point of the infection for at least 20 weeks, suggesting that 2.5 log of reduction in the viral dynamics probably enhance the life expectancy of these monkeys. Or if this is translated to humans, if this is same true in humans, it's going to increase the life expectancy. Again, the combination therapy is good, but what I am trying to tell you is here, although one particular therapy is not going to be doing anything for HIV, what we try to do is combining IL-15 with any LRA or any other immunotherapy like PD-1 or immune checkpoint inhibitors probably would actually give very good result or viral control for sustained viral remission. Again, I would like to acknowledge my people. I think Hema H. Babu in this slide, she did this experiment. She left my lab, and then all other people helped her. And I really thank my mentors here. Rama Amara is right, a collaborator of this in this one. Rafi Ahmed for a critical suggestion. Guido Silvestri, Paul, Chris, and Rafiq Sikali. And most every people have worked in this slide as well, helped me a lot. In fact, Rahman, who helped in the IHC one. Again, collaborators outside the US. Again, I'm a part of an Infectious Disease Society of India. We had a lot of discussions to really enhance the Infectious Disease Society. And again, this is a teamwork. This is a lot of people worked on this, and we are trying to do a single-cell RNA seq, as well as we are trying to get the reservoirs data to see whether whatever we found is actually true. And then just to take 1 minute to advertise this one, thanks for the organizer to letting me to show this one. So we are conducting a meeting in August, 6th and 7th in Chennai, and this is the second international conference on emerging and re-emerging infectious diseases, in association with the SRM, Institute of Science Foundation. And I'm a vice president, again, Amudan here, sitting here, he's an executive committee member in Immunocon, he's also general secretary there. We're trying to put together an infectious disease show. We did a great job in Chennai last year. Again, a lot of faculties from all over the world, almost 26 faculties all over the world coming and trying to bless us. Please, who are interested, please come and join. We'll have a good show there. Thank you so much.
This talk is open for discussion.
I'm sorry, I don't know. When I go close to it, it's giving a buzz sound.
It's okay. Any question from audience?
Hi, I have a question.
Yes.
So,
Yes, that's a good question. That's the thing that we didn't do. We are trying to do this experiment through a company, and it takes like 6 to 8 months to get the result. We gave it particularly the lymph node, we gave it to the company, but when we did this in the blood, we don't see any difference. Irrespective of what we see in the plasma viral load, it has not touched anything on reservoirs.
hi, Doctor, thank you. That's a great talk. I was interested, what is the route of vaccination? Is it all systemic or is it
This
mucosal?
subcutaneous.
But have you tried mucosal instead of subcutaneous?
No, I don't think. We didn't do that. Mucosal meaning in what way you were trying to deliver the cytokines?
So, long time back, I was working on HIV vaccines. It was based on a
Actually, this is not a vaccine. This is cytokines. One of the reasons, if you try to deliver cytokines in mucosal, it's going to create an inflammatory environment.
Aren't you using the cytokines as an adjuvant to your SHIV vaccine?
Yeah. But we, at that point, even though when we do it through adjuvant, when I don't know how they mucosally deliver both cytokine as well as the vaccine.
No,
but many people have did something.
I was
with
asking
DNA.
about the viral vector engineered vaccine. Is that done mucosally versus systemic?
No, we did another experiment and we followed up and we did the other experiment. In this case, we gave a vaccine IM, but we gave a cytokine sub-Q. That's a good question, but then it's a big question. If something we show inflammation, they're not going to let us to test the animals. But with the second part of the experiment, we also got a good results.
Dr. Velu, any question?
What is that? Right. So it's a very good question. So people, when they use IL-15 alone, it won't really enhance CD8 T cells or the NK cells in a very high level. But when you go administered together, actually it forms the dimer or whatever it is, but in that case, it helps to enhance high levels of CD8 T cells and NK cells. It has been shown, and one particular thing, it's called ALT-803. This is IL-15 treatment that actually been approved by FDA to go for a cancer therapy. So we did separately, in fact, these are monkey reagents, we did separately IL-15 alone and combination, the combination worked very well. Yeah, that's a good question. Sir.
Any more question?
That's right. That you are absolutely right. One of the reason, whatever we proposed was very high dose, but then we spoke to Louis Picker, who did this pioneering work on IL-15. He told me, Vijay, just try to put down the dose lower, because if you enhance a little bit more, it's going to touch CD4. It's going to activate CD4. Whenever you wanted to do a therapeutic intervention, particularly for HIV, you wanted to do something for CD8 and NK cells, the CD4 gets activated, you are done. There is no way that you can. We didn't do macrophages, but then we activated the monocytes. Actually, a good monocyte, that's the second part. I didn't show this. We activated these monocytes. Those monocytes help to CD8 T cells go to the follicles. Yeah, that's a mechanistic thing that we're working on. Thank you.
Sir, I have a question. Sir, given your previous work on PD1,
Yes.
nature work, so did you ever think of doing a combination therapy of PD1 and
Rama and myself submitted a grant a month ago.
Because
We've
that
been
would
thinking
be-
about it, but then this is the right time to hit that target. Yes, we are trying to do that.
Sir, and one more question is about the cost. So currently the cost of cytokine therapy is like $30K.
Yeah.
And what would be the duration that you need? Because right now we have the new generation of antiretrovirals, like biannually you give a shot and then the virus goes down. So how would you counteract it?
Okay,
Thank
so
you.
that's a good question. So since now the HIV field is moving towards the long-acting drugs, we are trying to do this experiment. We got an IACC approval. Laura's lab told me that they're going to give me the drug, but then one particular drug that they can't give. I requested Gilead, they're going to give. First one. So second, the very first question, what do you ask? The cost, okay. So if you would have listened to Rama's talk, we are trying to do something through RNA, LNP, or whatever it is, to deliver this one, particularly the L15. That's our alternative strategies that we are trying to do. But we wanted to look at the proof of concept to see whether it works combination with immune checkpoint inhibitors, particularly in this case, it's PD1. Yeah.
Thank you, Dr.
Yeah,
Willis,
thank you. Thank you so much for
your nice talk.
Thank you for Dr.
And,
Dil, Yazir, and
this
my good friend
cytokine
Amit Awasthi and the whole
therapy
team.
has a potential in controlling HIV and SIV infection. I have one question.
Yep, please.
In one of the editorial in 2018, I think, you have written about something, TGF-beta.
Right.
It also controls this HIV infection. Can you elaborate?
That's beautiful, Koser. This is one of the interesting questions that's been cooking in my lab. Okay, so there are two things here. If you look at chronic inflammation, in particular in HIV, when you look at Rafiq Sikali's lab, they published that the TGF-beta or IL-10 expand, they diminish antigen-specific cell. That's when you are not doing anything therapeutic. But what I feel is, in this particular experiment, what we did is, I don't have this slide actually. Did I? I don't have it. No, I don't have it. So interesting thing, what we did is in this particular experiment, we did this one, right? This one. So we sorted, we did a vaccine. We gave for an RNA-seq, blood RNA-seq to see what is signaling. Only TGF-beta stand out. Okay, now it's a chicken and egg question. People have published that TGF-beta is bad in HIV infection. In fact, I do have a grant that to put TGF-beta blockade to do something. But then, myself and Rama published a paper back in 2016 in PNAs. If you give TGF-beta, the CD8 T cells goes to follicles. They express CXCR5. But you have to have an optimum TGF-beta level in order to maintain the stem cellness of CD8 T cells. And I don't know, this is just, I've been observing this for almost a year. There are many people have started talking about this. Again, if you really wanted any CXCR5 positive CD8 T cells, that might go through this optimum level of TGF-beta. If this goes a little bit high, then there's a problem. And I'm trying to investigate that, but I just want to publish this first and then go for that. Yeah, that's really a good question.
Okay,
Thank
you very much.
Thank you.
It was a nice talk.
Thank you so much.
Thank you, Dr. Vijay,
Yeah.
for that compelling vision of cytokine-driven strategies in HIV-SIV cure research. May I now request our worthy chair, Professor Satya Das, to kindly felicitate our speaker of the evening, Dr. Vijay Kumar Velu. Please, sir. We don't have any lectures. No, please wait. May I now request our worthy chair, Professor Satya Das, to kindly felicitate Dr. Mahrukh Zargar, our co-chair of the evening. May I now request Dr. Mahrukh Hamid Zargar to kindly felicitate our worthy chair, Professor Satya Das. Ladies and gentlemen, we now move into one of the most anticipated sessions of Immunocon 2026, a brainstorming session of healthcare innovation and entrepreneurship, an interactive brainstorming session. May I request the chairman and the moderator of this session, Professor N.K. Mehra, to kindly grace the chair. Professor Mehra is former Dean Research at AIIMS New Delhi, Honorary Emeritus Scientist ICMR, and Founder Head, Department of Transplant Immunology and Immunogenetics, AIIMS New Delhi. May I also request the panelists of the evening, Dr. Vijay Kumar Velu, to kindly grace the chair. May I request Professor Rupesh Srivastava, Additional Professor, AIIMS New Delhi, to kindly grace the chair. May I now request Professor Amit Avasthi, President of the Indian Immunology Society, to grace the chair. May I request Professor Kaveri Srinivas to kindly grace the chair. May I now request Professor Saeed Mudassir Andrabi, Professor at the Department of Biotechnology, Sher-e Kashmir University of Agricultural Sciences and Technology, to kindly grace the chair. May I request Professor Clive Gray from the Stellenbosch University, South Africa, to kindly grace the chair. May I now request Dr. Ajazul Hamid, Scientist D at the University of Kashmir, to kindly grace the chair. May I request Dr. Muhammad Jamal Dar, Senior Principal Scientist, CSIR, Indian Institute of Integrative Medicine, Jammu, to kindly grace the chair. May I now request Dr. Richi Mahajan, Scientist D from the Department of Biotechnology, Government of India, to kindly grace the chair. Over to you, Professor Mehra.
And because this session has been made to discuss about industry-academia interaction. Most of the times the industry-academia interaction finishes at doing a MOU, or maybe one or two internships are done here and there. But I think there is a need for us now. We have reached a stage that the industry must develop in India, and there should be greater academia-industry interaction. I remember during the time, and as you know, I spent all my life in AIIMS, and there was a time that they will never allow you to interact with industry. When the Reliance Life Sciences was made, they included me as a member of their advisory board, and the institute said no to it. But I think times are changing now. Times have changed. And I want to give you one more example. When I was the dean of the institute in about 2013 or 2014, there was a call from Holland. They wanted to develop collaboration. There are 5 universities in Holland. They wanted to develop collaboration between AIIMS and those universities there, and their delegation came and interacted with us. They wanted us to also visit. So I selected a group of about 5 of my top really faculty colleagues from AIIMS, and we visited those 5 universities there. What we found was very striking. Each of the universities there was a new building there. That building, we asked them, what is this? I'm talking of 2013 or so. That was an incubator, a privately owned incubator, right inside the university campus. So there's a scientist who do any studies and they find something useful over there, and they can come and immediately do some interaction, and that's how this interaction develops. So I just made a couple of slides to highlight, and then of course I'm so very happy that there's a very worthy panel here. I was also keen to have one or two people from the industry in India, people who have manufactured any product in India. So when we talk of issues concerning industry, academia interaction in developing countries, there are a few issues that come up. One is a mismatch of goals and timelines. But academia runs on publications, teaching mode, grant cycles, success is measured in terms of papers, the PhDs graduated, peer recognitions, and timelines go on in years. The industry can't do that. Industry runs on a quarterly results, product deadlines, marketable products, cost saved, and revenues. And when you look at the technology levels from 3 to 9, academia works from TRL 1 to 3 only. A concept made, and we sort of signed an MOU. They don't go beyond that, and just one or two students over there go to the industry and interact. Whereas the industry has to go on to all those levels, 9 levels, make sure that the marketable product comes in the market. They can't wait for those 3 years or so or more. The second issue is trust and IP barriers. Universities generally lack tech transfer offices, clear policies on who owns the patents from joint work. Industry, on the other hand, fears leakages, and they fear faculty exploitation. Then there is a problem of confidentiality versus publish or perish. In academia, we only say publish or perish. Low risk appetite. Companies in low and middle income countries often like to import proven tech to co-developing certain technologies. They don't have the means actually of doing it. So they just import the technology from outside and start selling it here. Then the issue of capability and infrastructure gaps there. Outdated curricula that we have. The courses lag 5 to 10 years behind the industry needs. We really need to really activate our academia in terms of our courses there. And this is true especially in AI, the biotech, and advanced manufacturing. And labs may lack equipment for prototyping. We need to scale up and compliance testing. Then there's a talent drain. What we are training in our universities is these graduates who get trained over there, they often migrate to either multinationals or more often abroad. They just go over there and they feel very happy that they've gone up. Today, the focus is on startups. As you know, the Honorable Prime Minister has already, all the time keep stressing the point that in 2014, we had only 100 startups over there. And yesterday, of course, Govardhan Das said that today we have 168,000 startups in India, more than half of which are actually now in biotech industry, medical industry as well. But how many will go forwards? So there's issue of funding. There's issue of incentives, which is misaligned. Look, basic research is preferred over translational work, even today, and from lab and market. So there's a mismatch between industry and the academia. Consulting over co-creation. The industry would like to hire you as a mentor or as an advisor, but they don't want you to be a partner for the R&D. And then there are all those problems of bureaucracy there. I want to highlight one or two more points. Communication and cultural gaps. Different languages we speak. The industry speaks a very different language from the academia. Academia speaks in theory, citations. Industry wants specs, margins, performance indicators there. limited exposure. When we are in academic universities, the academia rarely does industry sabbaticals. And therefore, the Honorable Prime Minister said last year, we would like to pay for this. Let the sabbaticals be done in the industry. Industry R&D heads rarely sit on academia boards. Have you ever seen those industry big heads come and be on your board? Geography clustering there, all of this. I really don't want to spend too much of time because the organizers have given us only about 45 minutes and I've got a huge panel overhead. Then there are policy issues, ecosystem weaknesses, fragmented schemes, weak intermediaries, and there are challenges for those SMEs, small and medium enterprises over there. You know, I have been, all my work and all my life in AIMS has been translational work. We were doing those testings and all that. As we do the test, we want to bring it out into the patient care. But nobody at the time guided us that you could even create an industry, something like my other colleagues in the US were doing. Most companies in the developing countries are SMEs, which lack bandwidth, capital for multi-university. So our issues are, the core tension is not in unwillingness, it is that the two systems are optimized at different goals. Our goals are not the same. Bridging the two needs translators, advisors, risk-sharing funding. Our industry, our academia rarely does this risk-sharing funding there. And the career paths that eventually can merge. PhDs in academia are trained only to have their papers, and what we need is that they must have industry exposure. I was giving my own example when I retired from AIMS in 2014. I was in industry now. Of course, I was both in academia as well as in the industry. And when I learned that the industry has a sales team, it's a huge sales team, they have a huge marketing team. I was trying to create a center of excellence in HLA and transplant immunology in SRL lab. And they came to me, sir, we need a marketing team, you need a SRL team, in the sales team. I said, I don't need that. They said, no, you don't. And after 2 or 3 years, I understood, yes, there is a need for that. So those are the kinds of things that we do not teach our students. And in the next 2-3 minutes, just want to give you one glimpses of the industry that I'm now involved with. I'm involved with a startup where we are trying to create cell and gene therapy. It's an industry where we are trying to create cell therapy for immunotherapy, actually, trying to create CAR NK cells and CAR gamma delta T cells. And we are focusing only on 4 cancers there. But you see how you move. You move from an idea to the cells that you want to have. You need to generate huge amount of data. And this company has already passed 4 years. In 4 years, we have come to a stage of animal trials. And our product seems to be extremely good, and we are very excited. And the product on the data shows, and therefore the BIRAC has given us a grant of 45 crores to set up the GMP grade facility in Gurgaon there. There is still a lot of road from the data to the hospital where it can be used, where this product can be used for caring, and access. Everybody would like to have an access there. So the journey is not linear, it is an evidence ladder there. We need to have the clinician view, startup review, discovery phase, CMC process, preclinical, regulatory entry, clinical. There are lots of those steps over there. That is why it takes time. 4 years have passed, and we are now at a stage that we can feel that we come up. So when you're looking at cell and gene therapy, you're looking at viable cells. Those are your drug. Viable cells are the drugs. And the product is equivalent to cells plus the vector and plus how you make. The problem is due to the vector over there. So potency is not one assay, and safety is actually layered. I don't want to spend too much of time on this, but I want to spend little time on this. The major regulatory hoops in India. Oh my God, this was a big sort of challenge in the last few years. You need to have clearances, and there are several, from the IBSC, the biosafety point of view, RCGM, the DBT RCGM, CDSCO, IAC in the clinical side, GMP grade. You need to create your own GMP grade facility. And mind it, the cost, the minimal cost comes to be about 60 to 70 crores there. This is where our startups are failing. Startups don't have that much of funds actually. And then you finally come to the marketing stage. It's not an easy process there. So your mindset has to change from what you learned in academia and how you can translate it into industry there. So maybe I'll stop here. I don't want to go on and on, but I'll keep standing here because the panel is all full. I'm so happy. Maybe I start with you, Serini. You are from France, so you are an Indian French. Yes, you have the Janda of India as well, and also the France. So the issues that I raised over here, any comments from your side?
Now it's on. It's on. So Narinder, I like the comment that you made in the beginning. In the very beginning you said, why do you want to work with the industry? 45 or odd years ago, when I went there, one of the first things that I heard was, it was almost like a taboo. If you work with the industry, or you are a scientist, what are you doing with the industry? You're supposed to be a pure scientist, no? You're not supposed to. No, it changed very quickly. Okay, on a personal note, as a CNRS scientist, I am evaluated on the basis of how much I work with the innovation, how much I work in collaboration with the industry. In that sense, I have been lucky because, as I mentioned a few minutes ago, I work on intravenous immunoglobulins, and which is a multi-billion-dollar business, and the poor industry. There are a lot of people, a lot of laboratories who are working on that, who are producing, but they do not know fully the mechanisms. And when we propose to them, we will study these mechanisms in our lab, they're extremely happy to collaborate. That's my personal note. Now, coming back to the French system, French people, although in the beginning they were, talking of money, exactly contrary to what happens in the United States. In the United States, you start talking first money, how much you make, and then how much you make is important. Otherwise, you are not important. Okay, if you are making good money, then you are worth talking to me. In France, it's exactly the contrary. Yeah, it's the contrary, meaning you don't talk money. Money is not that important. But now they are insisting. The French system has developed very well. It has developed so well that there are many mechanisms. I will not go into the details, but very quickly, all the French ecosystem, one or two quick words about French ecosystem. French ecosystem is somewhat similar to India. We have CSIR, we have CNRS. You have ICMR, you have INSERM. You have ICAR, you have INRAE. Okay, I can go on. The beauty of the French system is, I am a CNRS scientist. I work in a hospital, in an INSERM unit. My students come from university. My colleagues are from the industry or from anywhere. That flexibility is the most important thing. L'Oreal people who are working on dermatology.
Maybe 2 decades ago, all your students were with the academic background.
Yeah.
Today the scene has changed. A lot of students are coming. They're supported by the industry, by the way. Yes.
Bilkul.
Their fellowships are being supported by the industry.
Exactly. Thanks to IVIG
Because,
once again.
in India, the problem is you get a student, you don't have a fellowship for him to be there.
Exactly.
And here the industry can do that.
Exactly.
This is the kind of change that we want in India.
Absolutely. And
I am not talking at the institutional level, something that AIMS does or Kashmiri University does.
Yeah.
My title was Industry Academia Interaction in Developing Countries.
Yeah.
So the problem is almost the same over there. I gave you the example of Holland.
Yeah.
You see that country. I've worked over there for many years. I mean, I used to go every year for a month or so and all that. Small country, and I used to tell my Dutch colleagues that your country is a 2-hour country. You stand in the center of the country, drive your car 2 hours, this, that way, you are out of the country. Yet, the biotech industry in Holland is most well developed over there. Why are they getting that? That's the example I gave you of the 5 universities over there. They are spending huge amount of money on that, the incubator, private incubator. The private and public partnership is right there on board.
Yeah. One last word about the,
yes.
Let's not forget, France is a country of Louis Pasteur. And the moment the Pasteur and the Institut Pasteur, they started developing vaccines, there was an industry. Immediately, Pasteur vaccine or Pasteur vaccines started more than 100 years ago. And the Pasteur Institute started coming all over the world. There is a Pasteur Institute there, Pasteur Institute here. The idea was obviously to treat the infectious diseases with the vaccines. That's first. But they were making vaccines, which means there was a commercial link to that.
So this issue of academia, industry partnership can't be more relevant than in the field of immunology. Because immunology is one field we have to move from basic to now translational. There are a number of products that can be made. I never thought I will get into this industry thing. I'm not an industry thing. I'm mentoring them and asking them. But the problems that we face in India. I'm actually going to be having a session with the government officials, including the Mantriji, very soon, to tell them our experience. What have we gone through? That slide I showed you, the number of clearances that you have to get. It's not an easy job. Now, I don't want to pick name and all, because the panel is really very big, and I don't know how much time we have. I have the organizer right in front of me, and she would probably show me a red flag sometimes. Let me know. I want to see anybody wants to speak first on, and then I will pick. Who wants to go? You want to talk about it? Yeah. You have the Emory experience?
Yeah, I do have an Emory experience, and when we found the PD-1 technology. At that point, when Amit might know about that, there when we started to tell something that we patented, the investors came just like that. Is it the same trend here? Just asking.
No, it's not. It's not.
Take the mic or
yeah,
you
so
move your chair forwards a bit so that everybody can hear you.
When we started this technology, when we said that we have patented some technology that could able to cure something, but the investors came very fast, particularly in US. Recently, I'm involved in a company where within a year or span of year, investors came and then they wanted to sponsor close to $5 million. Is the same true in India? That's my question.
No, it is not true. It is not true. Because you know that almost you have to approach the industry. It is not the industry you will come to your door and just ask about your technology. So it is not possible. So there are basically a lot of hiccups. There is a wide gap between academia and industry. So this gap need to be bridged. If exactly we want to translate a technology to the industry, absolutely, the industry there should be completely tied between academia and industry. So there is that gap is here. So that is not.
Okay, thank you.
Anybody else? Yes. Please introduce yourself. I don't know you very well, so please introduce yourself.
So I'm Ajaz from University of Kashmir. So I was just, I think, is it on? Yeah. So I just have a comment about distinct capacity building for the university versus academia, because most of the academic institutions, we train PhDs obviously for the academia. So there is obviously that lag and disconnect between the PhDs or graduates produced from the academia so that they can go directly to the industry. So given this sort of lacuna there, so I was wondering whether, like we have MD, PhD programs, whether there can be something like PhD programs which can train them or bridge the PhD students for the industry. I'm not sure we have it in India or whether we have such kind of programs in other countries.
Thank you.
Unless somebody else wants to come in here.
Ek minute. I'll get back to you.
You know, in France, there is a very clear mechanism where if the industry has certain questions that need to be addressed, and they don't have all the tools to do that, whereas an academic lab has. So they provide.
Srini, one point I raised on my slide, and that is my experience, and of course very true from others. Industry, when you get out of academic institution at a later stage, they want to hire you as an advisor, mentor, but not necessarily as a R&D partner. What we need is R&D partner while you are still in service. I gave you one example. Professor Tarasaki tray that we use for HLA typing and all that. He was a big professor in UCLA. While being in the academic line, he created this company, 1Lambda. And today 1Lambda is #1 industry. Anyway, you want to go first?
So, I'm Dr. Rupesh from AIMS Delhi. So what I believe that to do that, the best would be to change the PhD setup. I think China has already started up. They have abolished the system of publishing, and instead they have started off at least having one innovation, one patent, and get a PhD. So that could be a nice start for India also. If we can follow these principles, that will be really great. Instead of having this kind of conversation between an academia and your industry, if we start right from the PhD itself, have an innovation, get a PhD, and that's the best point to have this. So we have to maybe imbibe that system into the Indian PhD ecosystem also. Yes. So that could be one thing. And the government is also trying that. For example, the BRIC ITH program is there, the new PhD program, that is having that kind of 6 months of training in the industry, having their problems, and then imbibing that and then doing the research. That's one thing. Plus also the Prime Minister's Research Fellowship is also there, where the private industries are actually contributing 70% of the corpus for the PhD setup, for the PhD fellowship. So that is, things are there on track. But we need to be more fast if we want to be a really leader or pioneer in this. Yeah, thank you.
I know you are here. Don't worry. You know, the Prime Minister, of course, there is so much being done, but and we all know that. But in the road to create a product in India is very bumpy. Still, it's not only in India, it is in most of the developing countries. I want to turn first to a very sharp young guy, Dr. Amit Awasthi. He worked for several years in Boston with Dr. Vijay Kuchuru, as you know, and Vijay has interacted with the industry in a very, very large scale. In fact, he has established a big institution. In the industry, if it gets allowed, in academia-industry interaction, industry will come forward to create a center for you. All the money. At the moment, we are all the time dependent on government funds. How much funds can the government can give? They can't. And unless we come to a stage that 70% of your funding in research comes from industry and 30% from government, our biotech industry in India will not grow. Amit, your point of view.
Thank you, sir, for giving me this opportunity to deliberate on this. I think the system, what we see in India, is still not fully mature, and I think this happens anything what we start. Initially, we see a lot of hiccups, because if you really see, we have to balance two things in order to really achieve what other great nations who are like scientifically doing good achieved. We have to keep balance between the fundamental science and the industry or industrial research, because we cannot really punish the fundamental science saying that everybody has to do translation. What you are going to do in this process, somebody who trained to think in a different way, and now suddenly you're asking to make a product and give me a product in 5 years. That's not going to happen. What you're going to do in this whole process, you're going to kill your fundamental science, and you will not achieve the output what you will achieve, because we are not trained to produce the product in a kind of settings what you have mentioned. You have multiple layer of regulations. I sit in some of these committees in the CAR T-cell guidelines with the CDSO. We are trying to frame the guidelines, which is kind of, and one of the major problem in the CAR T-cell or cell therapy, what you have very clearly pointed out, that they are asking that RCGM, that you have to have GMP grade product, 3 batches. And then if somebody, I have a startup, right? I cannot have that GMP facility with some amount of money what I have investing from my pocket, and trying to get the investor, which they come later stage. And I had raised this point to the CDSO, but still they are kind of not really sure if they're going to change. But the other thing, what I think is the right thing, what I think should be done, we have to find a right partner. Like we do in a marriage setting, right? You have 2 people who are agreed to live together. And
just,
that's what
I want to stop you for a minute.
industry.
Dr. Andrabi, there are a lot of good points coming up, and you have been very kind to interact with me and help me as kindly keep noting down some of these points and the bullet points here.
I hope I am audible. Yes, yeah.
You want to speak?
Yes, yeah.
I'll call you. I'll call you. Yeah. I know you are here.
Okay. So, first, name is Mohammed Jamal.
No, I wanted Amit to finish his point.
I just quickly finish. So I think, let's say the environment in the India right now for cancer immunotherapy, not only necessary for the CAR T-cell therapy. 1 in the 10 people are going to develop cancer. And keeping the population size, you can imagine, is going to be more than the infectious disease in coming times. Because as we are going to develop, we are going to have sanitation measures, we are going to have clean water, and we go away from all this infection, and what ultimately leading to us, type 2 diabetes, cancer, and all these non-communicable diseases. So now I think this is the right time for India to invest, thinking that, our population is actually aging, and then age-related complications, which create a lot of burden on the GDP, need to be start investing at this point of time, so that in future we are kind of capable of handling such problems.
Yes. Thank you. And you raised a very important issue. I really liked it, and I know that it is a problem. The mindset change. Even our expert group members, I've seen the expert group members. If I go or you go with this industry thing at the back, they think, "Pata nahi kya kar rahe hain ye log." Something is fishy. They always suspect you. That suspicion and that mindset has to change. Our expert group members, they have to also, rather than pinpricking here and there, be supportive to the idea, because the other issue is you get this CGM approvals and they want you to have the GMP grade facility. Yes. Our company had to struggle for about 8 to 10 months, to get the GMP, and luckily sort of we got the grant. Yes, please. You are from DBT, right?
Yeah, no, I am from CSI Laboratory, Jammu. My name is Mohammed Jamal.
Your name is?
Mohammed Jamal Dar. I'm senior principal scientist there. So, we're talking about the mismatch. I was hired some 15 years back. So, with the idea that we are a drug discovery institution. Okay, am I audible now? So, I'll just give one example of mismatch. It so happens that if I remember 15 years back, we decided when I was hired that I was hired in a drug discovery project, and we started developing anti-cancer drugs. So where I was 15 years back, I am at the same place now after 15 years. Partly, what happens that when there is a clinical issue, we identify a disease, and then what is the primary requirement basically? When you identify a disease, you have to go to a cell biologist or a cancer biologist who will deeply understand the issue, clinical issue. Then you go further to a structural biologist who will identify the target, the protein, which you have to target. And then you have to identify a medicinal chemist who will develop small molecules to target that protein. And in between, there's a lot of bioinformatics involved. So that's basically the primary requirement for developing a drug in a clinical setting, in an industry, or in an academic institution. But if we go 20 years back or 25 years back, when we started developing institutions, we primarily trained our students, PhD students, for academic activities. Our idea was that we will give them training, they will go for postdocs, and they will go for higher education. We never trained these students for drug discovery. Yeah. And then when they were trained, they were put in as assistant professors, and then they became associate professors, and then they became professors. So their primary training is not related to this one. Yes. And suddenly our director or somebody else starts up developing a drug. He puts those people in this.
Thank you.
In this setting.
No, you made your point.
This is a mismatch, because they don't even understand what to develop and how to develop.
Thank you very much.
Compulsion is there.
Thank you very much. I want to go to my friend over here, young friend. He has been raising his hand for some time. And Clive, you will have the last word. Yes, please. I can't give my mic because I have to keep moderating.
Hello. Yeah. So I'll introduce myself. I'm Dr. H.E.V. Mahajan from Department of Biotechnology. I'm the program officer for BRIC, as well as some other flagship initiatives. I'll just go word by word what has been discussed over here, and I'll just try and address, because many of the things are policy based and all those things. We were around $10 billion industry back a decade back. We are marching towards $300 billion. We are approximately right now $162 billion. So we should at least appreciate ourselves that we are actually moving in the right direction. One thing. Secondly, government.
We are definitely moving in the right direction, but the road is very large. $160 billion for a country like India.
I totally get that, sir. Right now,
we don't have to be happy about it.
I totally get that, sir. Right now, probably we are around 3 to 4% of the GDP by industry, which is, and we should be around 9 to 10%. But that's the vision actually we should have. I'll come to the next thing. Department has recently, or I must say government has recently marched towards biomanufacturing scheme, which has been launched by the Department of Biotechnology. BioE3 policy, which was launched by Honorable Prime Minister last year. And it definitely actually boosts this.
1 minute. How many of my young colleagues in the audience are aware of the BioE3? Oh, quite a few. Very good. Very good. Yes.
Okay, so I'm very happy that you people are aware of the policy.
DBT should be doing something more to popularize BioE3 and how our young friends can benefit out of it.
I mean, we're constantly doing our webinars and outreach activities, but definitely point taken, and we'll reach out to everybody, every nook and corner of the country, so that the country is actually bio-dependent economy rather than the techno-dependent economy. Dr. Rupesh was talking about I3C program, one of the program which was conceptualized during my presence in BRIC. This program has one unique feature which is actually not available in any other PhD program, and that's 3 months immersion-based program, wherein we actually give 50,000 per month to a student, and we don't ask for any document or anything. We just tell them to go to the field and understand we don't even want the result from you. We definitely are looking for a report where he needs to understand this problem of the society. So I think Amit would be aware of it.
Tell them also how DBT supports the youngsters to do a sort of internship in industry.
I'm coming to that, sir. That's my next point. That's my next point. So that's what I'm saying, that we identify the partners for them, and we tell them to actually identify the grassroots level problem. Because, if being a scientist, I can even change a life of one single person around me, that actually adds to my value. Coming to your point, sir, we have recently launched EIR program, Entrepreneurship Residential in Residence program, where actually we are we actually kind of support postdoctorals or doctoral level fellows, and it's a brick by rack program where we've come out with the call, and there are industries mentors to them, so they can actually establish their own startups. I'll quickly wrap up on two more things. What we were talking about, we've been very complaining, and before taking DBT's job, I was fortunately associated with CDSU and drug regulation. So I'll just come to CDSU and drug regulation. So I won't say anything in in my defense or their defense, but we are a country where we call pharmacy of the world, and we called so because all around the world we are exporting around 190 countries, and we are trusted brand in the world. So because of these regulations and all these things, we are the trusted brand. So I think we'll have to actually, if we don't go into these kind of stringencies, and I totally agree, sir, that, and we had in 2016, we had biosimilar guidelines, which were there to ease the things. But if we don't go into these stringencies, there are a lot of people bypassing the things. There are a lot of large industries which are bypassing the regulations. I mean, trust me, I was part of the deal, and it's not easy to bypass. Last thing, I will just two things I would like to highlight. Many people are not aware that scientists in India are actually eligible to start their own country, in their own industry, even in service. The OM was issued clearly by DOPT as well as other ministries.
So this is something new. It
So
was
I can-
never there
I can
during
share
our
that
time.
OM with you.
Scientists or any academician, while working in university, is now allowed to start his or her own industry.
This OM was issued actually a decade back, but unfortunately it was not popularized. So, I mean, this is there.
एक बात मैंने डॉक्टर गोखले को कही थी कि 2, 4, 5 GMP ग्रेड फैसिलिटीज बनवा दीजिए. स्टार्टअप के पास पैसे नहीं होते.
Sir, I'm coming to that. Last point.
है ना? Last point.
Coming to that. Okay. Further, to facilitate the industry as well as academicians, we have actually relaxed upon our DBT IPR policy, where we don't now have any stake in the IPR. If it is developed by the academician, it is up to the institute as well as the academician to decide how to work upon. Like previously, they used to come to us what to do with the IP. We don't actually now interfere in it. The new IPR policy has been released, and that's notified on the DBT website as well. What you are referring to, sir, GMPs and other facilities. So DBT is actually coming up with a plan of having what you call bio parks. What you say bio innovation parks. We are actually conceptualizing. We are already had a Genome India project, which has generated a humongous amount of data. We are actually moving towards the second phase of it, where we are actually targeting more than 1 million genomes. So the thing is, industry don't has to spend on, a sequencing part. The data is available, work on it, drive whatever you want to.
Very good. Thank you for giving us an overview of what DBT is going through. DBT is now in its new avatar. Andrabi, I have to come to you later. I must give chance to Clive first. Clive has a very good habit of listening, and he has been sitting quiet. I want to know from you, Clive, what is the situation in South Africa?
Thanks.
And you have been coming to our conferences regularly every year, because you are like an Indian South African.
So I think, well, first of all, let me just say that I can also talk from the African perspective, and of course I'll talk more about South Africa, but the population of all of Africa is even a fraction of what there is in India. And I think that's very important, because the infectious disease and the non-communicable disease burden is proportionally higher because we have a smaller population. But within a South African context, there are quite a number of well-developed academic industry partnerships. We have the National Research Foundation, which partners with, these are governmental councils which partner with academics. Sasol, which is a petroleum company, is probably the most developed. it came out of apartheid, but it's now developing young, specifically black individuals to be in the market for developing innovative, not health, but innovative engineering solutions, especially around the mining. There's the Department of Science and Technology and Innovation. They included the word innovation recently to kind of embark on the mission that South Africa needs to compete with the world. And we do that through innovation. There's also the Universities South Africa collaboration, which tries to link and unite universities with industry. More recently, there is a company called Afrigen, which is the manufacturer of mRNA vaccines. And the quest there is that South Africa and Africa should not become, and this was born out of COVID, should not become reliant on the US or Europe. We do a lot of business with Africa, especially with the vaccines and the generic drugs. But needless to say, in my own impression and my own experience, is that there may be these wonderful partnerships. Two things, wonderful partnerships, but the students who benefit from those partnerships, and I think that's already been said, is they get absorbed into the academic and are lost from industry. So they become the professors. And they cease to become the innovators and because let's face it, professors are no longer innovative. That's where we need industry. The other is, and I want to be a little contrarian here, is that money is outpacing the welfare of humans. And whenever I hear partnership with industry, it's about money. Let's make money. Somehow, something has to shift. And especially, I think, on the African continent, and specifically in South Africa. My own institution, there's a particular department called Innovus. It's to promote the spin-off of ideas into companies. But the aim is to make money. We should not be making money at the expense of the health of our nations. Something has to change in our structure and our framework. So no one has said this here, so I'm going to say it. We cannot go this route. We have to change.
Thank you very much, Dr. Andrabi. Very short, because I don't think we have too much of time. Do we have any time? Another 10 minutes.
Okay.
Because I want to then open it for the house as well. Some questions from the audience.
Yeah, I have to attend one.
No, you can go. Yeah.
Okay.
My apologies.
Thank you for your very nice remarks.
So, I'm Professor Mudassir Andrabi. I am head of the division. Biotechnology, I'm heading the division. First, I will tell you that, talking about the startup. I'm having the startup. I'm the faculty, but I am running simultaneously a startup. And to narrate my story, that I have developed a first vaccine in the India. It is a Salmonella vaccine in the India that I gave to industry, and through the support of DBT, especially Dr. Nitin Jain, who was involved with the regulatory questions, and with the support of BIRAC.
Yes.
They helped. It is not like that. There is no agency that India that are not helping the scientists to translate the technology into the real world solutions. Exactly, they helped that. Secondly, coming to out, yes, of course, there are a lot of things. Why did these startups doesn't success? Only 5% startups success. There is 5% or less than success rate of these startups in India is very less. So my question is basically regarding that, because we don't have a clear mentor, whether it is at student level, whether it is at faculty level. To establish a startup in India, it is a very easy job. It is not something very difficult to task. But question is that in startup, what do you want to go? What do you want to do? How much money you want to invest and how much money you want to earn? We don't have that knowledge. And nobody is going to support you those questions to answer those questions. And you are the owner of the director of the startup, you have to just to resolve all these questions by their own. That is a big game I want to establish.
So I showed you a slide
yes.
on all of those steps. It's not easy. I mean,
yes.
you are saying that making starting a startup is not difficult, it's very easy.
Yes, of course.
I tend to differ on that because
yes.
I think
I will tell you, because myself as a faculty, I got a startup within 1-2 months. Registered startup. It is very easy job. But the question is that, can you run a successful startup? That is different.
Yes.
Okay. Now coming to the another point is that I have developed another technology. I have developed for a febrile fevers. I have developed a real-time based real-time kit. It is a real-time diagnostic kit. And what I have already cleared, that CDSCO, MD13 level I have already passed. I have already gone through the human clinical trials. Now we are validating that kit at the different primary care hospitals. But again, the question is that regulatory pathway in India is not so easy. Because myself as a scientist, how do I know the regulatory things? Nobody is going to support. Now DBT, especially I have seen, ICMR has those corridors. They are now have opened those corridors. But nobody exactly having the knowledge of those corridors, how to intervene. Because the persons who are there, they are not much qualified just to handle you or just to
I know.
They are not so
very good point. And I think what we are saying is that the universities and institutions should be advised to have a separate section.
Exactly.
Separate advisors and all that. That's fine. Anybody from the audience has any point? I hope you enjoyed the discussion here. This is something that we must continue in every conference. Maybe give more time to that. Yes, there is one. Yes, please. Oh, there are 2, 3. Please come forward. I think you should come forward because we don't have the mic and I can just pass on the mic from here. Get up and come right here and face the audience and ask your question. But keep looking at the organizing secretary. Once she says no, go for dinner, go. She's the boss. Why don't you come to the podium? Yeah, all of you. And please be very brief.
Okay. Good evening, everyone. I am Dr. Niaz, Assistant Professor, Department of Biotechnology. I have one query regarding one of the professors. He told that we should introduce PhD submission based on submitting patent. So would it be how far it will be possible in India to submit a PhD before completing a patent? So if the patent, it may take 10 years, it may take 5 years. So how can a student
Good.
go for
No, we understood. Rupesh, go ahead.
So yes, that's a very important question because we need to tackle that also. We need to do something which is actually not patent, but yeah, something innovation somewhere in between that. And we need to have certain regulations, certain guidelines, how we can do that. Because we are not going to follow that right now. But yeah, if we start thinking on this, we can have all these question and answers. We can formulate the guidelines so that students are not stuck, basically. Yes. So that's the main point. Because right now it takes a lot of time for a patent. We all know that. And that's why we also publish instead of patenting.
Thank you. Yes. Yes, Sirini.
It should not be patent oriented at all. The industry has a question, the academy has a question, we bring together, you answer the question, defend the thesis in such a way, and it is possible. We are doing it in France very regularly, where they are not published. If you think that the data are confidential,
Sirini, what is happening in India is students submit the thesis and then it takes about 1 year or even more for the viva to take place. This is criminal actually. And this is something that we have to improve. I think
we have to have the model of Holland. I used to go there very much. Thesis submitted, maximum of 2 months, and the report comes, and you don't have to wait for the convocation to take place. That panel, there is no convocation there. The panel who asks you the question, the examiners themselves sign and give the degree to you, so that you move on. Here, the viva doesn't take place for a year, and then the degree is not given to the student for another year. Thank you very much. Anybody else? Please, those of you want to ask, please come forward.
Good evening, everyone. I am Tanshu, and I wanted to ask, what are your current thoughts on backstabbing in economics? And further on that note, what are your current thoughts on trust? How can you trust someone in their financial state?
Who wants to answer that? I'm unable to answer this question. Thank you. Thank you. Yes.
Can you repeat this question again?
No, he said he said something about finances and all. Yeah.
Financial trust. Yeah. I am Professor.
That's what we are asking the DBT to open their trust of all the funds, you know. By the way, this young man, क्या नाम बताया यार बेटा? Nitin Jain है ना? हाँ. The DPT was started along with me, with Dr. Ban. I was the first person to get that time DPT was called NBTTB, National Biotechnology Board. I was the first one to get the research associate of NBTTB. I had not even applied. Ramalinga Swami was the chair and they gave me this. Yes, please.
Good evening for all of you. I am Professor Tippeswamy from Karnataka, Coimbatore University, working in Coimbatore University in the Department of Microbiology. So first of all, in the name of innovation or patent, just if you ask for industry to interact, and what I mean to say that industry expects money after the completion of the research, but the research student, if they start with innovation or patenting idea, it is not 100% sure that he will end up with patent or innovation product. But the industry comes only at the end of his research journey, whether it is working or not working.
Nitin is saying we are trying to create a risk fund,
yeah.
You know.
What I mean, so another thing is that in general, at least all the PhD students must get financial support, at least to stabilize themselves,
yes,
to think over it,
yes,
to think about the
yes,
problem.
Otherwise,
if you just pressurize them,
yes,
they cannot think innovative ideas,
yes,
and many patenting ideas, how to control the disease, nothing, because he will focus more on invention or
absolutely.
Product,
you made your point. Yes, we have understood your point.
Yeah,
thank you. Thank you.
Impact factor.
Madam, please come. Yeah.
Can I speak from here?
Yes. Thank you so much.
Am I audible from here?
Yes, you are. Yeah.
Okay. So I'm Madhu Tapadia from Banaras Hindu University. And as you all know, it's one of the largest universities. We cater to almost 12,000 students in science every year. So what I feel is the students in the campus, so there's a lack of, there's a disconnect between the industry and the academia, and we as academicians, we are not able to actually help them, because we ourselves are not very clear about how this interaction or the crosstalk between industry and academics works. And what I feel is now with the new education policy, if we can have certain courses which are focused, we have soft skills or skill-based enhanced courses, or the other courses value added, where the industry can actually guide us what kind of courses for a 6-month a student should take, and so that can help him to think of some innovative ideas, one thing. Second is, if campus placements can be, encouraged, if industries can come to the campus and they can actually tell what they want from the students. So
that's a very different thing you're talking about. That is already being done. You see in the IITs and all that. What we are saying is for product development.
No, so IIT
Industry,
is
academia,
IIT
interaction for product development.
what I'm saying is, for the basic students, science students, if the industry can come and tell them how the students can help them,
Yes.
you know. So what are the questions? So that I think if it's encouraged.
Thank you very much. I think that's about all. I'm sure everybody wants to go for the dinner now. Yes, please.
I am Dr. Nisar. I am teaching in a medical school of the Kashmir, renowned and a prestigious institution of the Kashmir Government Medical College, Srinagar. I am teaching from last 16 years there.
Teaching what?
I saw.
Teaching what?
I'm teaching biochemistry, sir.
Biochemistry.
Biochemistry. And I saw when our MBBS students come, soon after completion of the biochemistry, this MBBS, they start the internship and they are being paid a good amount. Then they pursue their FD, MS and other courses very comfortably. So they are financially strong and sound. Just now, sir came here and he took a very important point that we too in universities are getting the PhDs, but those PhDs are not comfortable as far as their financial strength is concerned.
Yeah.
Now, what is the solution? Respectively, I would like to request all dignitaries here who have a say in the central ministry that why we should not request the finance ministry to enhance the budgetary allocation for the university education so that the PhDs we produce, they will be sound, they will be financially sound, and they will
there
a
has
lot
been,
they can contribute.
I can
contribute
tell
a
you
lot.
in the
Thank
last couple of years or maybe 3-4 years, there has been a consistent increase in the allocation. The problem is on both sides. I also see that the DBT, DST-like institutions and all that,
Thank you.
they are also hard pressed. They want to make sure that the money that they get, the budget that they get the enhanced budget actually. But time comes when they don't know where to spend and how to spend. So I think there's a mismatch, but that goes on everywhere. Yes, please. I think we have to close very soon.
How much is the stipend that you guys give for the PhD per month?
As of now, I guess it's 37.
I think we should all be very happy with the COVID experience. You know, when COVID came, we had only one lab, NIV, and then within a matter of months, we were not even manufacturing the PPTs. We were not even manufacturing gloves actually. In a matter of months, really, the hundreds of labs were established. Not just that the labs were established, they were validated and all that. And all those equipments were being manufactured. So I think there is so much of talent here. What I highlighted in my slides was mismatch. There's a lot of mismatch between industry and academia, and we need to really close that gap. And I'm happy to hear that now the academia, scientists are allowed to start up their company as well. With that, I think I'd like to thank you all for your very wisdomful words and all that. I think this debate must go on. We must make sure that we जो मैं कह रहा हूँ आप कहेंगे
नहीं सर आपसे ही
You have a point?
Good evening, everybody. I am Sayyidina Mulhaq. I'm a PhD scholar. I'm a student of Professor Andrabi at SKICC, Kashmir. Well, among the distinguished speakers, someone rightly said that in the academia and at the policy level, we have seniors essentially who are of the mindset that, of the academic training. Now, problem lies there, according to me. If we start training our PhD students in an industry-wise manner, that would be great. Now, what I had an observation back in our university at SKICC, in our PhD program, in sync and in line with the new education policy, they devised that we had the advisory committee. We include one member that's of the industry. That was a wonderful thing because we had students sent across India into the industry. Now that there is the point where we start training our PhD scholars in industry your line or fashion, however you say it, but that they are the start of industry psyche training. I think those PhD scholars down the line in 10 years, 5 years would be at the policy level. So they would already have the training of industry setup and they would proliferate. Thank you. Thanks.
Very good point that the DCs, the doctoral committee of the students should have also one representative from the industry. That's a very important point. I want to stop here and I want to thank all my panelists here for their contributions. So you might like to now go on. Thank you very much.
Thank you so much.
Thanks everybody. Thank you very much.
Thank you so much, sir.
Give them a big hand, please.
Yeah. Thank you to all the panelists. We have a quick felicitation ceremony. Can I ask all of you panelists to kindly come to the front of the dais, please? First up, may I request Professor Srinivas.
This all should go one, and Madam should or I should.
May I request Professor Srinivas Kaveri to felicitate Professor Mehra. May I request Professor Narendra Mehra to felicitate Professor Clive Gray. Thank you so much. May I request Professor Dr. Muhammad Jamal Dar, our guest Professor Srinivas Kaveri. May I request Professor Saeed Mudassir Andrabi to felicitate Dr. Vijay Kumar Velu. Thank you. May I request Dr. Vijay Kumar Velu to felicitate Professor Amit Avasthi. Okay. May I request Dr. Vijay Kumar Velu to felicitate Dr. Muhammad Jamal Dar. Thank you. May I request Dr. Rupesh to felicitate Professor Saeed Mudassir Andrabi. Thank you. May I request Dr. Ejazul Hamid Wani to felicitate Dr. Rupesh Srivastav. Thank you. May I request Professor Narendra Mehra to felicitate Dr. Richi Mahajan. Thank you. May I request Dr. Richi Mahajan to felicitate Dr. Ejazul Hamid Wani. Thank you to all the panelists. Thank you so much all. Ladies and gentlemen, that brings day 2 of Immunocon 2026 to a magnificent close. From the eye-halt concept to therapeutic antibodies, from HIV cure strategies to the mid-career award, This day has been a testament to the extraordinary breadth and vitality of modern immunological science. We now move to our gala dinner, an evening of warmth, celebration and fellowship. Details are in your conference booklet. Please join us and enjoy the hospitality of Kashmir. Day 3 begins tomorrow morning. Please check your booklets for timings and hall allocations. Thank you. Thank you to all.